as a tribute to Mother Saraswati, the goddess of knowledge. Dr. Tanuj, please do the proceedings.
या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा। शुक्लां ब्रह्मविचारसारपरमाम् आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदाम् जाड्यान्धकारापहाम्। हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्।
Thank you very much. So, ladies and gentlemen, without further ado, may I request Dr. Anil Kumar, Director Education, to bring into the light the house with the theme and objective of this webinar through his introductory remarks. Over to you, sir.
Thank you, Dr. Nishant Bhanu. Our most distinguished chief guest, Professor K.C. Bansal, Secretary, National Academy of Agricultural Sciences, New Delhi, former Director, NBPGR, New Delhi, and former Professor, NRCPB, New Delhi. You being one of the highly recognized biotechnologists of the country, we all your colleagues feel delighted on your accomplishment for serving this great nation. We feel honored by your gracious presence today in this national webinar, sir. Another distinguished plant molecular biologist of the country and currently holding a position of Executive Director, Navi Mohali, and Professor Life Science, Jawaharlal Nehru University, Delhi, is the guest of honor and one of my close friends, worked together and authored books on biotechnology in agriculture and medicine, principle and practice. It is a moment of pride for me when our colleagues like you signing on national platform. Honorable Vice Chancellor, Dr. Arvind Kumar, sir, who is the founder Vice Chancellor and architect of 3rd Central Agriculture University and brought first institute of national importance at Jhansi as Rani Lakshmibai Central Agriculture University, Jhansi being a heart of Central India and also land of fiery queen of Jhansi. It is having magnificent building and infrastructure for imparting quality agriculture education under his steward leadership. The most important, our learned speaker in this webinar, Dr. Batri Vanthan Mani, Senior Scientist, Genetic Resource, ICRISAT, Dr. Swarup Parida, Staff Scientist 2, National Institute of Plant Genome Research, Dr. Kalyan Babu, Senior Scientist, ICR, Palm Research Institute, who is happened to be one of my PhD student and in the same platform in year 2016, He and me awarded Jawaharlal Nehru Best PhD Thesis Award, and myself has been conferred as Dr. C. Subramanian Outstanding Teacher Award from ICR New Delhi, beside curious participants from different states of the country. Our colleague from neighboring institution, Director IGFRI, Dr. Amrish Chandra, and Director Kafri, Dr. A. Arunachalam, Dr. S.K. Chaturvedi, Dean Agriculture, Dr. Anshuman Singh, Organizing Secretary, Dr. Nisant Bhanu, Convener, and other member of organizing team, Dr. Manit Rana, Dr. Piyush Bavele, Dr. Tanuj Mishra, and Dr. Sailen Kumar. Our colleagues, faculty member, scientist of RLBCAU,
आप म्यूट हो सर सर please unmute sir.
A very good afternoon and a very warm welcome to all of you in this national webinar on new dimension in crop improvement from allele mining to genome editing. This year we are celebrating 75 years of independence as Azadi Ka Amrit Mahotsav and also Vigyan Mahotsav 2022 as part of National Day celebration. In this important year of India's 75th anniversary of independence, we are organizing this national webinar on the most emerging, challenging, and exciting subject having enormous application in agriculture and health. Today, we have with us a panel of very distinguished scientists of par excellence. It is our wish to invite all of you in our university physically, but due to coronavirus pandemic threat, we could not do so, and invited you to interact with our stakeholder through digital platform in this webinar. We know very well that the genetic diversity is a foundation for crop improvement. Traditional varieties or land races of the crops harbor a large store of valuable genes and can be used to develop new variety with improved yield potential, higher nutritional quality, and higher tolerance of the stresses for future climate changes. The prerequisite of crop improvement lies on the availability of germplasm termed as primary, secondary, and tertiary gene pool, followed by hybridization and selection as important step in plant breeding. Methods are now available to evaluate the genetic worth of the vast genetics resources archived in gene bank and streamline application of these resources in crop improvement programs. High throughput phenotyping methods are beginning to alleviate the challenges of accurate, precise, and large-scale measurement of plant performance, and also use of optimized speed breeding protocols when applied with genomics breeding approaches. Beside allied variety, together with the land races, can produce pre-breeding materials with reduced linkage drag. The breeding methods can also be used to generate a gene pool enriched with potential traits. Multi-parent advanced generation intercross, that is called magic population, is a breeding method to produce high-quality recombined germplasm. The increased recombination in magic population can lead to novel rearrangement of alleles and greater genotypic diversity. Sustaining gains from genomic breeding seeks fast tracking exploitation of the minor effect alleles, accumulation of the favorable alleles, and purging of deleterious alleles. Over the past decade, genomics-assisted breeding has been instrumental in harnessing the potential of modern genomic resources and characterizing and exploiting allelic variation for germplasm enhancement and cultivar development. Now, we are going in the era of genomics-assisted breeding and sustaining genomics-assisted breeding in future, that is called genomic-assisted breeding 2.0, will rely upon a suite of new approaches that fast-track targeted manipulation of allelic variation for creating novel diversity and facilitate their rapid and efficient incorporation in crop improvement program, and will be indispensable for designing future crop. In coming decades, genomics-assisted breeding 2.0 is expected to play a crucial role in breeding more climate smart crop cultivars with higher nutritional value in a cost-effective and timely manner. Availability of the reference genome and genome-wide surveys on comprehensive diversity panel pave the way to associate the allelic variation with phenotype. The recent completion of resequencing of 3,000 gene bank accession, that is called 3,000 Rice Genome Project 2014, and other crops like chickpea has revealed the allelic diversity of multiple genomes. Genome and allele mining approaches have been made tremendous advance to dissect naturally occurring allelic variants of candidate genes with essential agronomic qualities such as resistance to biotic and abiotic stresses, nutritional quality, yield potential, and other. Multiple alleles from the complex locus serve as a reservoir of variation to generate functional gene. The routine sequence exchange such as mutation, recombination, gene rearrangement, and polyploidy are the main mechanism of gene evolution and development. It is now possible to dissect the actual allelic variant of the genes within an array of crop cultivar via allele mining. Allele mining can be an important method to identify additional alleles and new haplotype, along with the development of allele-specific marker for use in marker-assisted selection. It can be visualized as a vital link between effective utilization of genetic and genomic resources in genomics-driven modern plant breeding. Precise genome editing technology in concert with enhanced trait architecture enables innovative solution to engineer complex trait variation. This webinar, we are focusing on the actual concept and potential of mining approaches for the discovery of alleles and their utilization for agriculturally important trait in crop. New tools for the precise modification of crops genes are now available for the engineering of new idiotype. A future challenge in the emerging field of genome engineering is to develop efficient method for allele mining. In 2015, we have published a conceptual review in the Omics, a Journal of Integrated Biology, on CRISPR-Cas9 based genome engineering opportunities in agri-food nutrition and healthcare. Considering the growing importance of this particular subject, the various methods of allele mining have been followed, such as genotype by sequencing, GBS analysis for SNP detection. We have performed taking calcium as a trait in finger millet, and now several sequencing platforms are available for dissection of the complex trait by following the sequencing and resequencing approaches for SNP analysis, as well as also using phenomics and genomics platform. And it will really help for identification of all potential alleles using reference and test population. And such development are now making basis of genomic selection using SNP array. Genome engineering has been made remarkable development not only in agriculture, but also in health. In 2020, the Royal Swedish Academy of Science announced the Nobel Prize for the development of a method for genome editing in chemistry, and awarded two women scientists named as Dr. Allele Charpentier as well as Jennifer Anna Dodana. These are the two women scientists have been awarded the Nobel Prize in chemistry on this genome editing techniques. So at the last, I would like to say that genomic engineering tools are now available in plants, including major crops, to modify in a predictable manner a given gene and have a tremendous potential for a spectacular acceleration of the plant breeding process. The speaker shall discuss how genetic diversity has always been the raw material for breeders and how they have always taken advantages of the best available science to use and, when possible, increase this genetic diversity using genomic editing tools. Genomic engineering by site-specific nucleases enables reverse genetics and targeted editing of the genomes in efficacious manner based on clustered, regularly interspaced short palindromic repeats, that is called CRISPR, related RNA-guided endonucleases facilitate coherent integration of the crop genome function, and application of this targeted genome editing platform with the inherent potential to use basic science, strength, and shortcoming of gene function that will complement plant breeding techniques for crop improvement, somatic genome editing, rectification of genetic disorder or gene therapy, treatment of infectious disease, generation of animal model, and crop improvement. The application of such emerging tool for allele mining and genome editing in agri-food nutrition shall be discussed by our learned speaker in details in today's webinar, Professor K.C. Bansal, who has really worked on genetic engineering of plant and now emphatic as well as the chloroplast engineering, and now emphatically talking about the importance of genome editing in crop improvement. With this, I am ending my introductory thoughts about this national webinar. Thanks for your patience hearing.
Thank you very much, sir, for your honest introductory remarks. As you all know that the Rani Lakshmibai Central Agriculture University, a university of national importance, has been established in 2014 by an Act of Parliament under the Central Agricultural University Act 2014. Like other agricultural university, Central Agriculture University Jhansi has key objectives to impart education in different branches of agriculture and allied sciences, undertake research in agriculture, undertake programs of extension education, and promote linkages with national and international educational institutes. In a very short span of time, we have attained some of those goals, and we remain committed to our steadfast work to achieve the rest in due time. I would request our coordination team to kindly show the glimpse of the journey of our university so far. Dr. Tanuj, please.
लक्ष्मीबाई केंद्रीय कृषि विश्वविद्यालय की स्थापना वर्ष 2014 में संसद द्वारा पारित अधिनियम के अंतर्गत राष्ट्रीय महत्व के संस्था के रूप में वीरांगना रानी लक्ष्मीबाई के नाम से प्रसिद्ध झांसी में हुई। विश्वविद्यालय लगभग 300 एकड़ के क्षेत्र में विस्तारित है। ये विश्वविद्यालय न केवल बुंदेलखंड अपितु देश के विभिन्न राज्यों के विद्यार्थियों को गुणवत्ता युक्त कृषि शिक्षा, अनुसंधान व किसानों को तकनीकी प्रशिक्षण प्रदान करने के लिए प्रतिबद्ध है। साथ ही साथ यहाँ उन्नत बीज भी उपलब्ध हो रहा है। इसके साथ ही पशु चिकित्सा एवं मत्स्य महाविद्यालय की स्थापना भी इसी विश्वविद्यालय के अंतर्गत दतिया में की जा रही है। दलहनी एवं तिलहनी फसलों के अनुसंधान का ये विशिष्ट केंद्र है और यहाँ दक्षता युक्त शिक्षा भी दी जा रही है। इस विश्वविद्यालय के 2 नवनिर्मित शैक्षणिक एवं प्रशासनिक भवनों का निर्माण कार्य नवंबर 2017 से प्रारंभ होकर लगभग 2 वर्षों की अवधि में ही NBCC द्वारा पूर्ण कर लिया गया। शैक्षणिक भवन का कुल क्षेत्रफल लगभग 17,500 वर्ग मीटर है, जिसमें 19 स्मार्ट क्लासरूम सहित विद्यार्थियों के लिए 23 शोध प्रयोगशालाएं भी उपलब्ध हैं। इसके साथ ही ये भवन सर्व सुविधा युक्त बहुउद्देशीय हॉल एवं सेमिनार हॉल से सुसज्जित है। प्रशासनिक भवन लगभग 5,000 वर्ग मीटर क्षेत्र में निर्मित है, जिसमें समिति कक्ष एवं बोर्ड रूम जैसी सभी महत्वपूर्ण सुविधाएं उपलब्ध हैं। विश्वविद्यालय में कुलपति, कुल सचिव, निदेशक, अधिष्ठाता के साथ साथ लगभग 14 राज्यों के उत्कृष्ट शिक्षक निरंतर अपनी सेवाएं दे रहे हैं। इन भवनों में ऊर्जा दक्षता के सभी मानकों को समाहित किया गया है, जिसमें सौर ऊर्जा पैनल, LED लाइट, वर्षा जल संचयन, तकनीकी एवं प्राकृतिक संसाधनों का प्रयोग किया गया है, जो इस विश्वविद्यालय को प्राकृतिक स्वरूप देते हैं। माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी के कुशल नेतृत्व में जारी नई शिक्षा नीति से कृषि शिक्षा के क्षेत्र में भी युवाओं का कौशल विकास होगा और स्वरोजगार के अनेकानेक अवसर प्राप्त होंगे। आज इस विशिष्ट विश्वविद्यालय को माननीय प्रधानमंत्री राष्ट्र को समर्पित कर रहे हैं, जिससे हम सभी गौरवान्वित अनुभव कर रहे हैं।
Thank you, Dr. Tanuj, for this. Now, I would like to invite Dean, College of Agriculture and patron of today's program, Dr. S.K. Chaturvedi, sir, to share his words of wisdom and guide us with his experience. Over to you, sir.
Good afternoon, everyone. Honorable Vice Chancellor of the University, Dr. Arvind Kumar ji, world-famed researcher and research administrator, and today's chief guest, Professor K.C. Bansal Sahab, Dr. Parekh Sahab, I can say Dr. Beshampayan Sir, Dr. Sarup Parida, Dr. Sahai, and my own colleagues from neighboring IHR institutes and from the university, my dear students and friends. I joined Dr. Anil Kumarji in welcoming you all on this occasion. As the tone has already been set by Dr. Anil Kumarji regarding today's webinar, that's on new dimensions in crop improvement. I would like to share some of the things of my views just to set the tone so that the London speaker can take off. With this, as suggested that I should share something about that sustainability for future, for self-sufficiency. So what really it means that the sustainability can be visualized with the four aspects, maybe the social issues, human related issues, then economic benefits and environmental benefits, or in terms of economic and environmental issues. So rightly, just I thought that pulses are the best bet because my background is on pulses research, so I thought it's the best bet also for the sustaining self-sufficiency of food grains and also to ensure the social, human, and economic benefits to the country at large. So sustainable production base, certainly pulses are required, nutritional security, then the ecological security. So pulses are the best bet. That's I feel. So really what we mean by sustainability. The sustainability in terms of social, when we say to preserve future generations and to acknowledge what we have done or what we are doing and what will be the likely impact of our activities on others as well. Means here we have to respect the equality and for the Panchabhuta. What for human, as we know that natural resources and spaces are limited. So certainly balanced growth is required with improvements in health and economic well-being for one and all. Means we have to suppress our greed. Until we suppress our greed, we are not going to really achieve the goal of sustainability. Then in terms of economics, what I had said about the social, same way is here. Then to maintain the capital intact and to improve the standard of living, means maintaining high and stable levels of economic growth is one of the most important aspect of the sustainability. Fourth pillar is the environmental one. So human welfare through the protection of natural capital means the respect to Panchabhuta again. So we have to really take care of everything. So what really has been done so far? How to really explain the self-sufficiency? So if you see the production of food grains over the period of time in our country, started just I thought that let's compare with the 1950-51 just to have better understanding. Just if you see that in case of wheat, as on today, means I have just compared with the data of last year. So if you see then something like 17 times increment has been achieved in this country in terms of the wheat production. So we know that wheat, rice, and in wage, so these crops, Green Revolution was realized in early 70s. So the contribution of wheat was tremendous. And if you see even on today, the rice production, it has gone up by more than 6 times. Same way is the maize. A tremendous improvement has been achieved in case of maize. And if you compare with the 1950-51, then more than 18 times increment has been seen in production of maize. Of course, pulses were lagging behind, but during last few years, pulses have done a good job. And if you say that instead of producing 8.5 million tons, now country is producing more than 25 million tons. That the more than 3 times increment has been reported achieved in case of pulses. Coming to the total food grain production, again, more than 6 times increment has been realized. This, as on today, we are self-sufficient, at least with respect to cereals and pulses. Why I say that? Because the demand for the pulses has been estimated by various researchers and the planners, something like 25, 26 million ton, and by 25, the 27.5 or 28 million pulses will be required. And I am of the opinion that with the kind of positive policy support of the government and the technology available, certainly in case of pulses, we can sustain the self-sufficiency also. Of course, whatever products import are there in case of pulses or in other crops, those are the due to the our trade or the business. We have to develop the friendship, so we are importing as well. Otherwise, we are now self-sufficient in case of most of the cereals and case of pulses as well. Then we have achieved the pulses revolution, as we all know, and that with pride we can say that the pulses revolution has been achieved through indigenous technology developed by our own researchers, and the production has been achieved by our own farmers. So what are the really challenges and dimensions of the sustainability? Let's discuss something about this also. So the growing population, that's the biggest challenge, and we have to feed the ever-growing population of this country. Then the increase in income has taken place. So certainly with the increase in income, demand for quality food is also increasing. Health consciousness is increasing day by day. So the requirement for healthy food is also increasing. Employment-led social changes, like the in most of the cases, husband, wife, let's say both are working or the entire family is working. So they don't have really time to cook the food. So in that case, demand for ready-to-eat food is also increasing. Then the manpower for agriculture is becoming expensive because its shift is there from agriculture labor to the industrial labor. So certainly a lot of pressure is there. Then the how we can achieve the really the sustainability goals or the related pillars. Like for social, what I feel that we have to produce more, ensuring the affordability of the healthy foods. Then that will ensure with that we can ensure the food diversity if we are producing more. So certainly the food diversity through nutritionally rich food has to be there, and for that pulses, wild seeds, and millets are the best bet. On economic front, we have to produce more, higher productivity or profitability. It means we have to reduce cost of cultivation as well. Then the environmental, how to really sustain the environmental, how to look at the environmental issues. So crop diversification is the best bet, and where the inclusion of pulses in cereal-based cropping system particularly has to be ensured so that we can sustain the agricultural production base as such. Then just to apprise you that today's webinar is related to the crop improvement, new dimensions. So certainly something I have to share about the historical background. As we all know that crop breeding just started with the introduction of germplasm and through by acclimatization or a selection, then the purification of land races, and later we started the resorting for the hybridization, and many varieties have been developed through simple crosses, through mutation breeding, and at later stage composite, synthetic, and hybrids were developed and released, and they are at the farmer's field. So so many things have been done during last 60, 70 years. Of course, during 1960s, the major breakthrough for development of infrastructure taking place when Indian government initiated program on the name of All India Coordinated Research Project. Once those were established, starting with the maize and then followed by other crops, then sharing of germplasm and breeding material increased. Then the breeding program started moving from merely selection from the land races to the simple or to multiple process. And hybridization also came in picture to broaden the genetic base of selected crops. Then at later, the molecular markers were developed by many researchers. Dr. Bansal was pioneer in that, and many researchers, they resorted to develop in this particular area. And then the breeders also, what they did, they also realized that as usual business may not serve the purpose, so they started uh, including the traits which are being preferred by the farmers and the consumers. So number of varieties having the farmers and consumers preferred trait were also developed. Then the accelerated breeding. Earlier we were using the oxygen generation advancement. Now the techniques that the speed breeding, where we can go for the seed to, we can multiply seed. So seed to seed cycle can be achieved just in few months. Like in crop of say crop of chickpea or in wheat, in these cases, 4 to 5 generations can be completed in a year. That has really that may help in near future. Then the transgenic and gene editing to create new variability has been launched, and I am sure that our learned speaker will be discussing more about these things. Then what these all breeding programs or the integrated breeding program, I should say, with the help of the particularly crop physiologists, plant pathologists, entomologists, agronomists, and many scientists, they joined hands, and then country has developed more than 3,500 crop varieties. Only 344 varieties were released during last year of various field crop, and most of these varieties, they have the resistance against the major phytic stresses, that the insect pests and the diseases. In case of abiotic stresses, drought, waterlogging, heat, and salinity, so number of varieties are now available for many crops. Then the special characters like the early mature, high input response, and farmers and consumer prefer traits like extra large or large seeded varieties, then the nutri-dense varieties, then the amenability to combine harvesting in case of pulses and in other crops to minimize the use of agrochemicals. So many cost-cutting traits were incorporated in the existing varieties, and now we have excellent varieties in many crops. Then during last 2 to 3 years, more than 2 dozen biofortified varieties have also been released. And through popularization of these varieties, we can ensure nutrition security to the people of this country. Then where is really the opportunity to sustain the self-sufficiency? So the power rests with the pulses, because pulses can fix the nitrogen, and they can open up the soil to the deeper strata. They can help in breaking the deep and compact layer in the soil. So certainly, pulses are the best bet, and they offer a lot of opportunity. Why I say that? That we have developed a number of varieties in this country where the duration of crop has been reduced. So certainly, between two cereal crops, pulses can fit, like the mung bean, bean. There are many pulses like cowpea. So these 3-4 pulses can fit very well in that cereal-based cropping system. So those can help in crop diversification. Same way, we have reduced the maturity duration of chickpea, lentil, pigeon pea, field pea by something like, let's say, 15 to 30, 40 days. In case of pigeon pea, tremendous work has been done. Now, instead of having the 240 days varieties, we have just varieties maturing in less than 120 days. So such type of achievements have been made in past. Then the extra large seeded cowries have been developed. Then the lentil varieties have been developed. These varieties are creating wonders, and they offer a lot of opportunities to diverse for the crop diversification, and particularly from the rain-fed area. These crops with the minimum irrigation can be popularized. Then in case of mung wheat, as I said, early maturing varieties like maturing in 50-55 days have been developed, and these varieties have the simple maturity. So certainly, in a small window, these varieties can be fitted well, and certainly the agronomic base or the agricultural production can be improved. Then we have now very good variety in case of field pea as well, that has the green seed, so that can be used for diversified food. Same way, the number of varieties having the biodiversified traits like the iron, zinc have been developed. So these all varieties are helping in crop diversification. Only the support from the policy is required. Then the top research priorities of the globe should be the nutrient acquisition and use efficiency. Then the accelerated genetic gain at farmers field we have to ensure. Then the biofortification. It's not only for the human being. Biofortification should be for the crops as well as for the fodder, so that the health of human beings and the animal can be ensured. Then the cost-cutting technology. So the breeding and biotechnology we have to really consider. So why we say that cost-cutting technologies are really important? Just to quote example of the import of the fertilizers. Under this high input agriculture practices, we started importing fertilizers, and just see the example where most of the nitrogenous, phosphatic fertilizer, potash and sulfur we are importing, and government is providing huge subsidy on this. So just to take the example of the import, it's in billions. It's not the small amount, and huge fertilizer import is there in this country. Just to see that in 2020-21, country has imported fertilizer of worth more than 71,000 crores of rupees. So our import of fertilizer is increasing day by day. So what we have to do, we have to resort to some of the genetic potential of variety or the germplasm. For that, what we feel that enhancing the nutrients acquisition and use efficiency has to be done. So for that, we have to develop the soil resources where specific nutrient deficient soils can be developed, then phenotyping can be done, and we can select the contrasting traits, and then the mapping population can be developed, QTLs can be identified, uses fulfill can be identified, controlling this nutrient acquisition and use efficiency, and certainly the later varieties having the better nutrient acquisition and use efficiency can be developed for the future, so that dependency on import of fertilizer can be reduced. Then the improving the human and animal health, what we feel that arresting protein malnutrition and the nutrient malnutrition, that's the of utmost importance. So for that, the crops like pulses, oilseeds, cereals, and pseudo cereals, they offer many nutrients. So certainly we can have the we can popularize and we can develop the varieties suitable for different growing environments of various pulses, targeted pulses, oilseeds, cereals, and pseudo cereals, and those can be popularized. Then we are arresting the nutrients. Already I have said that for nutrients malnutrition, agronomic manipulations of the options are also available. Means the cultivation of the targeted crop on nutrient-rich soils or with the supplemental nutrients that can be done. And second can be the genetic option where we can develop the nutrient-dense varieties with the enhanced level of targeted nutrients in grains or straw as per need. Then improving the quality of produce to meet the consumer's preference. So day by day, as we said, under high input agriculture, farmers are heavily or indiscriminately using fertilizers, insecticide, pesticide, and other agrochemicals. So now the health issues are increasing, and many rich people, I should say, upper middle class and rich people, they are demanding for the quality food. So certainly, lot of scope exists to go for the organic farming. When I say organic farming or natural farming, I don't mean that the entire country should go for that, but certainly 10 to 15% land can be diverted towards organic farming or natural farming. Then the quality seed requirement. That's again important aspect for sustaining the self-sufficiency. So for the major crops, we have to develop the standards again. We have to we should have we know how to ensure the nutritional quality and the seed quality. So for that, to assess the genetic purity, I think the molecular markers can be utilized to replace the grow out test. What we do in seed certification, we go for the grow out test, and grow out test take not less than 5 to 6 months to confirm whether the seeds are genetically pure or not. For that, if we can develop the variety specific molecular markers to assess the genetic purity of a particular variety when seeds are seed requirement is voluminous. Certainly, with confidence, we can say with minimum time that okay, this whether this variety has the this particular seed lot has the quality or not, or how much admixture is there in that particular variety. So, assessment of the quality, genetic purity, or the quality of seed being produced by various agencies need to be done. So, for that, molecular markers can also help. So, many techniques and tools are now available. Then the what I said that there is need to go for the utilizing the or harnessing the potential of the powerful technology. So what I feel that rapid generation advancement technology is already available. So on the name of the speed breeding, now facilities are also available at many centers, and ICRISAT has recently opened that facility. And those who are interested can certainly use that facility that's available at ICRISAT. Then the identification of these QTLs, my colleagues, senior colleagues, Dr. Bansal, then my friend like Dr. Parekh and Dr. Parida, they shall be discussing about that. Then the deployment of molecular markers for enhancing selection efficiency in breeding program and assessing the seed quality or the genetic quality of the seed lot. Then the technologies for mechanization of cultivation. These are the now tools and techniques are now available. Only thing that we have to need some policy support, and with that policy support, certainly we can achieve what we wish to have. And just to summarize, that demand supply need to be reassessed for various crops. Then the bringing food diversity by way of developing ready-to-eat food, that's important. Then the synergies need to be harnessed for crop diversification. We need better varieties. Then setting research priorities for the crop improvement program. Then now the tools and the available tools and techniques. Now certainly we can go for the enhancing the efficiency of the crop improvement program. And I will certainly suggest to the young researchers that they should embrace these technologies so that the rapid genetic gain can be achieved. So with the higher production from per unit area of the cultivation, with the reduced cost, will ensure more availability of quality food at lower price. With increased affordability, that will ensure sustainability for self-sufficiency, because now the tools, techniques, varieties, and GAPs are available for most of these crops. And I am pretty sure the kind of support we are getting from the government of India, our farmers will be able to produce more food with sustainability. And this country will see the sustainable self-sufficiency in years to come. Thank you. Thank you.
Thank you very much, sir, for the illustrious speech on sustainability for self-sufficiency and highlighting about the achievements that have been made and the issues that needs to be emphasized to achieve and sustain this self-sufficiency. Thank you once again, sir. Moving forward, today we have with us the chief guest of today's function, Dr. K.C. Bansal, Secretary, NAS. Before I welcome him for his talk, I would like to request Dr. Maneet Rana to please give the audience a brief introduction about the chief guest. Sir.
Thank you, Dr. Nishan. Good afternoon to all of you. It gives me immense pleasure introducing Professor K.C. Bansal in front of you in today's national webinar. Professor Bansal obtained doctoral degree with gold medal from IRI New Delhi. He pursued advanced research from Harvard University, Cambridge, USA. Since his return from USA, he occupied eminent positions in ICR and served as Director, National Bureau of Plant Genetic Resources, New Delhi. Professor Bansal is an accomplished scientist of international repute. Currently, he is the Secretary of the National Academy of Agriculture Sciences and member Board of Directors, Global Plant Council. He was the first to get selected for the prestigious Norman Borlaug Chair for Crop Improvement by the ICR in India. He has significantly contributed in the area of plant biotechnology and plant genetic resources. His research areas include functional genomics and transgenic development for abiotic stress tolerance in plants. Professor Bansal has supervised over 20 doctoral students and imparted training to over 300 scientists at national and international level. He has published over 150 research articles in Journal of International Repute, including Nature Biotechnology and Proceedings of National Academy of Sciences, USA. He was elected as Vice Chair from Asia for the 15th regular session of the Commission on Genetic Resources for Food and Agriculture of the United Nations. Professor Bansal has won several awards. To cite a few, the Rafi Ahmad Kidwai Award, the highest award of the ICR for an individual's contribution, an award of the Indian Science Congress conferred upon by the Honorable PM of India, Biotech Career Award of the Rockefeller Foundation, USA, Recognition Award of the National Academy of Agricultural Sciences, and the prestigious Haryana Vigyan Ratan, highest state science award. Recently, Professor Bansal was awarded the second highest award of the National Academy of Agricultural Sciences by the Honorable Minister of Agriculture, Government of India. Now I request Professor Bansal for his guest lecture on genetic diversity, genomics, and genome engineering for crop improvement. Sir, please.
Okay, thank you. Thank you, Dr. Manit, and thank you, Dr. Nishan. Before I go further, I thought I would better share my, I'm disabled to screen my slides. Can you please allow me?
Yes, sir. Allowed now. Allowed.
Am I allowed now?
Yes, sir. Yes.
Can someone confirm that my slides are opening?
It's coming, sir. Yeah, it's visible now, but not in slides only. Yeah. It's okay, sir.
It's okay now?
Yes, sir. Yes, sir.
Okay. Good afternoon to all of you. Dr. Arvind Kumar, my very good friend and noted oilseed scientist in the country, and the Honorable Vice Chancellor of this Rani Lakshmi Bai Central Agriculture University, the founder Vice Chancellor, Dr. Amresh Chandra, the director of the neighboring ICR Institute, also Dr. Arunachalam, Dr. S.K. Chakravarti, the Dean Agriculture, Dr. Anil Kumar, Director Education, my fellow speakers, firstly, Dr. Ashwini Parikh, a very well-noted scientist in the area of molecular biology in the country and heading an institution of national and international repute, Mohali, my other fellow speakers, participants, faculty and students of the Central Agriculture University here in Jhansi, my dear friends and ladies and gentlemen. First of all, I would like to convey my sincere gratitude and gratefulness to the organizers of this particular national webinar, particularly the Honorable Vice Chancellor, Dr. Arvind Kumar, Dr. Anil Kumar, and Dr. S.K. Chakravarti, and all the staff of the university. I'm pleased to be here with you today, and it's a very important topic on which this national webinar has been organized, by the university authorities, particularly Professor Anil Kumar, who has been doing a great job, as I know, as the Director of Education. And the topic which I'll be talking to you today in the next about 40-45 minutes is on genetic diversity, genomics, and genome engineering for crop improvement. I'll be giving major emphasis on genetic diversity. At the end, of course, I'll be touching upon genomics and genome engineering, particularly for the sake of students, inspiring them to utilize these technologies for crop improvement, in addition to national plan bidding, which has already played a very big role. And why I'll be talking about mostly genetic diversity, because not only in India, globally, this diversity which is available to us, to the readers, to other scientists, has been, actually speaking, largely ignored. We must try to find value in this genetic diversity for any such program, whether we're talking of genomics, structural genomics, functional genomics, or identifying various alleles, or through various approaches of improvement, including even conventional breeding, or I'll be talking about genome-assisted breeding or genome editing, finally. So I think it's very important that we try to realize, and I would particularly request the younger students, the faculty members, those who are yet to initiate or looking forward to utilize the kind of wealth of information we have, not only in India, elsewhere as well in the world. I'll give you a little bit over that, and they must try to utilize that fullest advantage, not only in their own research, but also for the benefit of the country. Government of India has particularly set some of these priorities, whether we call them as government priorities, which are kind of outcomes, or even research priorities. If you look into the government priorities we want to produce more from less for more, or more, we produce more per crop, actually, or per drop, we produce more per drop, or we produce more crop per drop. Then we need self-sufficiency, as Dr. Chakravarti very rightly mentioned, not only in cereals, we already self-sufficient, but we need self-sufficiency also in oilseeds and pulses, more importantly, pulses. And minimum use of chemicals is advocated by the government of India, so as to save resources, naturally occurring resources, whether it is fertilizers or irrigation, land, or all other natural resources. And also the goal, as you all know, by 2022 was or is at the moment doubling the income of farmers, particularly the smallholder farmers who contribute to the 80% of our number of millions of farmers we have in our country. And more serious issue is from the government perspective is to tackle malnutrition. And this all only can be done if we can direct our research and we identify research priorities, particularly I'm talking of in the area of agricultural sciences. For example, we must try to increase productivity rates. Productivity rates, I mean here is per tons or productivity per unit area, okay, or per unit time. We need to increase that. And we need to accelerate the pace of this increased productivity or genetic gains by developing improved varieties. We need to develop varieties which are of shorter duration so that they utilize less resources and produce more. And we need resistance for these emerging pests and diseases. We need, again, tolerance and resistance to the various abiotic stress factors. Also, we need to incorporate a rate of herbicide tolerance. We need to worry about nutritional and processing quality, to tackle nutrition. And I said before, we need to increase the natural resource use efficiency. And finally, biological nitrogen fixation again, which Dr. Chaturvedi very rightly mentioned in case of pulse is important, particularly to bring sustainability in our agricultural production. And apart from all this, in fact, we have to achieve all this while keeping in mind that India is a signatory to this fact, and we need to meet these sustainable development goals of the United Nations by 2030, which is less than 10 years from now. And this slide I'm particularly showing to the younger colleagues, to show you the difference between the yield and production of cereals and pulses. You can see here, cereals production is way high. You talk of wheat, rice, maize in general, you can see here this is way high than in case of whether you talk of productivity or production in case of pulses. It is very, very low here compared to cereals. And there could be some reasons for it. And this is what, in fact, I'm going to lay more emphasis on my presentation, to pulses. Of course, cereals remain important, but then I will be focusing more on, for example, rice or wheat here, and of course, pulses, particularly chickpea, pigeon pea, or lentils, and including the work which we have been doing while I worked at NBPGR, and also, of course, including the work of some of my colleagues whom I would acknowledge at the end from NBPGR or from elsewhere. Now, just to make a point here very clearly, that how much reduced genetic diversity do we have, for example, in different crops, and this reduced genetic diversity is a potential threat to food and nutrition security in the long run. I mean, this is a slide which shows in case of rice, for a country like Sri Lanka, which was growing about 2,000 varieties of different rice about 60 years ago in 1959, now they're growing less than 100, or 100 is also a large number, much less. In India, we don't have, I believe, as many as we're growing currently, varieties of rice, though we have large diversity, which I'll share with you later. And 75% of is all, even if those 100 descend from a common stock, while, I'm talking of a material which is utilized by plant breeders to improve those crop varieties. Also, in case of Bangladesh and rice, 62% of the varieties descend from a common stock. In case of Indonesia, 74% of varieties descend from a common stock. That's a message it gives very clearly that we lack diversity, genetic diversity in rice, whether you talk of Sri Lanka, Bangladesh, or Indonesia. And also in wheat or in potato and soybean, it is a similar situation. Particularly, you can see in wheat, 50% of the crop is represented by 9 varieties, half of it. 75% of potatoes is represented only in 4 varieties, and soybeans is represented by 6 varieties, 50% of it in the United Nations. So that very clearly shows, that there is a lot of decreasing or decreased already diversity, genetic diversity in the farmers' field, particularly I'm talking about it. And this is another elaborate example in case of wheat. That in case of wheat, as you all know, we have these different wild species of wheat, with A genome, B genome, and D genome. And while this is a kind of a diversity which is shown here, in case of dots, like it is 100%, 10 dots, 100% and 100%, if we presume it is there in wild species, it got reduced because of domestication, particularly in A genome and B genome. You can see it got reduced. The number of dots you can count here is only about 30, it is now 70%. And it got further reduced when the hexaploid wheat, came into being through a process of natural hybridization, combining all the three A, B, and D genome. The diversity came down to only about, you can see here, 20%, what is available here, 100%. Each dot you can see is about 10%. So, 20% diversity coming from, only A genome or B genome and D genome, which was a process of natural hybridization occurring in nature, which gave rise to this hexaploid wheat, which is a hexaploid, with three, genomes A, B, and D diploid each, 2x, it becomes triticum styrene as the hexaploid wheat. But then fortunately, again, the hybridization took place between these hexaploid wheat and some of these tetraploids shown here, and land races were formed, which were having now increased diversity compared to the hexaploid wheat, which occurred as a result of natural hybridization. But further, through the process of artificial selection, here the systematic plant breeding, when it started, which of course gave rise to green revolution, no doubt about it. But you can see here the diversity from here got further reduced. Whether you talk of hexaploid wheat, which is or durum wheat, which is the tetraploid, it came down to almost, let us say, about 20 to 30. It was left only about 30% of diversity, here, and which is much less than we compared when you talk of wild species, or when you talk compared to the land races. So this is another factor that by nature it got reduced, it got increased further, but then artificial selection or the conventional plant breeding further played a role in reducing this diversity. Continuing with wheat, now if you compare different countries, that what kind of a diversity they have, in terms of the released varieties or the area which is being grown under different varieties. Now these are different sources, whether they are CG-centered developed cultivars or CG-centered derived material, or they are non-CGR material, particularly in case of wheat. So CG-centered here is responsible for it is CIMMYT. No doubt CIMMYT is working for international good, a wonderful piece of work. But in case of China, you can see here, most of the varieties, what they're growing are from non-CG-centered related material. That means they're using their own diversity or their own local kind of land races which are available, in China. And of course, less CGR derived and very less cultivars directly, which they would have released from the material nurseries they get from CG Center. But you see here in India, the situation is very alarming. In India, about 65% from CG cultivars or CG derived cultivars. If you combine, it becomes about 85% of this material. And our own effort from our own germplasm resources, if we have at all utilized them in our breeding programs, this is only the tip of, you can see here very less, contribution of non-CGR related varieties. So I don't want to go into the other countries, but you can see globally, if it is 50-50, 50 CG cultivars or CG derived cultivars is 50%, 50% is their own, if you say globally, but India, in fact, is, they are utilizing more of this. So that's another reason of actually speaking that we need to diversify, and I will give you those examples, what could be the reasons. Now, in case of pulses, a wonderful analysis done by Dr. Shivakumar, of course, about 20 years ago, wherein they analyzed, the pedigree analysis they did of 229 released varieties, and which revealed clearly a narrow genetic base in most of the pulse crops. For example, if you see in case of chickpea, out of 86 varieties which have been released, 35% contribution to these 86 variety is from only top 10 contributors as parental material in a breeding program. And out of these 10, top 5 ancestors are listed here. And this is the most important column, if you see here, which is the most frequent contributor, of the genes, or which is the most frequent ancestor which has been utilized in breeding programs. In case of chickpea, it is PB7, and which has been utilized 40.17% out of the total released variety in this 86. In case of lentil, it is 17%, only one, again, ancestor which has been utilized. Pigeon pea, 2 of them, contributing 34%. Wood bean, only one contributing 64%. And in case of mung bean, it is again 34, 35%, and it's only one parent. or one ancestor which has been utilized in breeding programs. And therefore, message is very clearly that due to the repeated use of the same germplasm, for the development of cultivars, there is a reduced diversity, as you can see, at a narrow genetic base. So what is required is that shows that a vast majority of accessions, or a message given here is, held in the gene banks have not been utilized or have made limited contribution in development of the modern cultivars. So this is, I talked to you about rice, wheat. This is, in case of different pulses. And also, if you look into the utilization of plant genetic resources by CG Center, the world-known CG Center, International Rice Research Institute, their own study they have carried out of about 100,000, 1 lakh accessions they conserve in their gene bank. Only 11% have been utilized as parent in the breeding process, only 11%. And finally, in the release variety, the contribution of this germplasm is only 2%. You can see here it's very minimal. You can imagine what will be the figure of 2% when we have got 100,000, and we have the similar kind of a number in our own gene bank. I will talk to you a little further when I go into it. So by this, what you would have seen by now, that there is a limited diversity which has been utilized. Diversity is present, but which has been only limited fraction of it has been utilized in breeding our crops. Now, but let us have a look at what is the global status of these plant genetic resources in the world. We have about total of 7.2 million accessions in 1,750 gene banks across the world. And who are the major contributors? Which crops? Cereals contribute about half of it, 48%. Food legumes, fortunately, is the second, in the line, and that is 16%, followed by other crops. And here, of course, it's a similar situation, which is there, but important is this. And where are these genetic resources conserved globally? These genetic resources conserved, I mentioned before, in 1,750 gene banks. But again, majority of them is also conserved actually in many of those international CG Center gene banks. I mentioned to you, for example, we have in India, ICRISET, and where of course, mostly the pulses, the millets. We have ICARDA, again, pulses, and wheat and other crops, ICARDA. Then we have, in Philippines, Manila, we have IRRI, Las Banos, IRRI, which is only for rice. Then we have CIMMET in Mexico for wheat and maize. Like that, we have got, of course, potato in SIP Lima, and we've got CIET. And of course, we have Biodiversity International. We earlier called it IPJURI, with headquarters in Rome. They have got on-farm, conservation of many of those resources out of 1,000, out of about 7.2 million accessions conserved in this. And they are shown here again. The yellow dots are shown are the CG centers, but others are the other, major, national gene banks, including we have in USDA or in Germany and some of them in Europe. But apart from all this, you can see a dot here near the North Pole, which is a gene bank with safety deposits in the Svalbard, seed vault, we call it. This is a Svalbard, seed vault, which is located, actually, this is the figure, located near the North Pole. You can see here it's all covered with snow all the time, or sea, which of course, at times get frozen. And this is that, kind of the only international gene bank. The idea is here to have safety deposits, forever. Forever for posterity. It means it is so important, these plant genetic resources, that the whole world come together, and develop this. Of course, it's funded by Norway, government of Norway, and it has been, and it comes under, I think, the jurisdiction of Norway. Geographically, it is different, but it is being funded by mostly the government of Norway. And this is, we call it a Svalbard Global Seed Vault. And I was fortunate to deposit some seeds of pigeon pea accessions while I was in NBPGR for the first time, on April 9, 2014. So this shows that how much importance we have, and we're trying to conserve these genetic resources. And this is just to give a glimpse of different types of germplasm which have been conserved across these 11 CG Center gene banks of different crops. I don't want to go into the details of it, except you can see here rice, of course, Africa rice, in addition to International Rice Research Institute, we have got about 1,24,000 accessions. And then we have got biodiversity, CH, CIMMET, all these are there. And these are number of crops which are dealt by these CG centers. Of course, we have otherwise, as you know, so many crops, germplasm conserved in all those national gene banks, across the world. And we have our own national gene bank, which attracts a very high level of importance, I would say, nationally, internationally. And we have total about today 0.45 million accessions, 4.5 lakhs. This is an old figure, about 4.5 lakh accessions of different crop groups. We talk of cereals, we talk of millets, we talk of pseudo cereals, we talk of grain legumes, particularly highlighted here, about 57,000 accessions, oilseed, vegetables, fruits, and all of the crop groups, primarily for food and agriculture. We conserve here in NBPGR, and apart from the seed gene bank, which is the largest, of course, and it is the second largest gene bank we have in the world after USA, we do conserve material as in vitro bank or cryobank, and also in the field. So this is a kind of a message now I want to give you so far what I have talked to you in those slides, that what are the lessons we have learned so far. With regard to crop genetic diversity, lessons learned or issues of what? That we have not fully characterized our material, or we have not evaluated. So first message is poor characterization in evaluation of plant genetic resources, and that's why it has not been utilized by the plant breeders. It's not a fault by the plant breeders. I would say it is also to do with the people who have been managing these national gene banks. It's not only, by the way, true, I'm talking about for the Indian National Gene Bank. It is true globally. I've visited, fortunately, several of those, meetings and some of those gene banks, and I find the situation is the same globally. The poor characterization and evaluation of plant genetic resources, irrespective of what crop we're talking about. And therefore, poor utilization of these resources by breeders. And also, of course, I mentioned before, there's an over-reliance on international nurseries. I give you an example in the case of wheat. Then there is therefore finally a narrow genetic base of some of those released crop varieties. I gave you quite a good amount of evidence, I would say. And this is the decreasing crop diversity is mostly, because of the spread of monoculture, only few high-yielding varieties contributing, as I mentioned before, 4 to 5 to the 50% of the total, crop area, or hybrids, of course. And there now GM crops have also come into picture, if not in India, and many other crops I'm talking about it. And people claim that there's a further loss of biodiversity due to GM crops on the farmer's field, particularly I'm talking about it, not, as such. Of course, the diversity has been conserved, even if true because of these high-yielding varieties. All those land races, certainly were not preferred by farmers anymore. But those were, as you all know, were collected and are conserved. I gave you a picture not only in India, but in other national gene banks of the world as well. So what is the way forward? Way forward to encourage the utilization of this genetic diversity is that we undertake systematic characterization and evaluation of the entire gene bank collections. In fact, this is something I was very happy, and I took it, myself when I had an opportunity, and never it was otherwise before that thought of, that can we try to characterize and evaluate the entire gene bank collection of a particular crop. It's not easy, but then we don't have time. We must try to utilize whatever the genetic variability is available in any given gene bank. And finally, you develop a core or a mini core or trait-specific reference sets and share that kind of characterized material, evaluated material with well-developed traits with plant breeders and researchers. And not only this is the simple characterization evaluation part of it. Then you have to broaden the genetic base by another technology, we call it as even, pre-breeding, when you can utilize these land races or clockwise relatives, and then using also in modern genomic approaches, you can try to, then enhance, the discovery of certain useful traits, and that's what is called basically genetic enhancement of that material, which then becomes a starting material for the plant breeders to utilize in their breeding programs. So this has also been very, has been utilized, this kind of the technology of the material to a limited extent, while there are efforts, I know, in different crops like wheat and rice in some of the organizations in the country. And also we need crop diversification with respect to nutritional enhancement. Not only that we always depend on wheat and rice or a few other legumes, I think we need to, crop diversify more on it. I'm not going to not talk much on this, but then also finally, the way forward is that we try to adopt GM root, which is very important. And also we have now genome editing technology is also very important. And it's not going to reduce the diversity. Rather, my message is very clear that it's going to promote biodiversity. And therefore important is the plant genetic resources, the genomics at root, the pre-breeding, and GM crops and genome editing, all need to be combined together, when we talk about, crop improvement of various crops. So when I had an opportunity, I mentioned to you, friends, with the discussion with my colleagues at NBPGR and several of the breeders, including even Dr. S.K. Chakravarty, I believe, through a meeting of all India breeders, we came to a conclusion that we need to we identified 15 of these crops, including cereals or millets or oil seeds and pulses and some of the fruits and vegetable crops. And we decided what are the important characteristics we would try to characterize or evaluate them, whether it is for resistance to biotic stresses or abiotic stresses, nutrition quality, or even earliness, photo and thermo insensitivity is also very important to diversify the area or increase the area under cultivation, and also for even hybrid seed production, it's required in some of the crops. And therefore, I decided, but during the tenure, I had an opportunity. I could do something with rice, wheat, and chickpea. And I will start with actually wheat. We had about 22,000 accessions of wheat, narrowed down out of about 30 because we removed duplicates. And we took this exercise in 2011-12, where all 22,000 wheat accessions grown on one field in HAU Hisar campus. And idea was here what? The idea was that we develop a core set, which is very important, which is a 10% of the large number, and which will capture about 90% of the variability, so that we are about only, let's say, left with 2,000 accessions, which is then a manageable number. So we develop a core set, which we did at Hisar, as shown here in this Indian map. Then we, identified germplasm for resistance to rust and powdery mildew, and that was done actually, in Wellington and in Cooch Behar, in North Bengal. And then another set of the similar number we grown at Isapur, that was to develop a reference set against terminal heat stress tolerance, which is near Delhi, as shown here. So quickly, I will give you an overview of all this, that we developed a core set, particularly to start with only one species, that is Triticum aestivum. Germplasm accessions conserved, and we found a good amount of variability, and this work was appreciated, in an international meeting, which was held in Japan in 2013. And we see here the growth habit. It is kind of, covering the whole ground, and this is erect and very less chlorophyll. And this has got a lot of waxiness on the leaves here compared to this broad leaves. And this is only, I'm showing you a sample. Otherwise, there's a lot of variability. Imagine if you're growing 22,000 accessions, which are all genetically diverse, by the way. So therefore, we developed a core, not only with Triticum aestivum, then we developed a composite core, comprising Triticum aestivum, Triticum dicoccum, and Triticum durum, and all the three species. And this was again published in Crop Science. And then finally, we identified material resistance to rust and spot blotch. Sorry, I can't give you details. I'm just giving an overview. And if someone is interested, can go to these papers or talk to me directly. We identified, minimal number, manageable number, which are resistance to rust and spot blotch. And similarly, we identified new resources, which are resistant to powdery mildew. This is again, in crop science. And then we finally developed a reference set, which is much in demand, particularly for terminal heat stress tolerance. It's very important, and we did it on these 5 different parameters, and we have got this number out of those 22,000, about 3,000, which are highly tolerant. And in fact, out of these 3,000, I'm not showing that slide, we have about 48 which are not at all affected when you talk of any of these parameters, particularly when you talk of grain yield. They are not at all affected as a result of heat stress, particularly terminal heat stress. So well, this work in fact got a lot of attention, and I'm very happy about it. For example, in cover Crop Science, it was specifically with this message came onto the cover, and with the link of these 2 papers, and then also ICR, put it into Limca Book of Records, and also this was the first trial which was carried out actually in speaking Wellington, which was picked up by a prestigious newspaper, as you all know, Hindu, and highlighted, this kind of effort, carried out by NBPGR. And Dr. Jaiswal, I think, is also there in the audience, who was there at that time visiting. I could not visit myself. But also, I'm very happy. I was inspired. I must tell you this clearly, and I want to clearly tell this message to the youngsters. When these scientists, they came to know, for example, our own Director General, Dr. Mahapatra, he says, excellent. It is a milestone, a landmark contribution. I'm talking about the collection, or I'm talking about this characterization evaluation of the entire collection of a gene bank. Okay, the kind of wheat I'm talking about it. Then Dr. Deepak Pental, great piece of work. Nobody in India has ever dared to undertake such an activity on germplasm. Professor Sudhir Sopori, another well-respected scientist in the country, internationally we have. This is a result of your commitment and passion for doing good science with application in our agriculture. Dr. Padhi, by the way, is an IAS officer. He's a country director of research, but a technical person. He's a PhD. He says, very proud to know about your work that would positively contribute in development of climate resilient and disease-resistant wheat varieties, which are very important. So we have the material identified now. Therefore, he makes this comment. And of course, our own good friend again, Dr. Ashok Singh, he himself is a very well-noted breeder. He mentioned it's indeed a historical paper, and I will circulate among my colleagues so that you can utilize the material more in their breeding programs. And that was our idea and the purpose. So in fact, that gave us encouragement. That was not only in wheat, but also we did about 15,000 accessions, but that was done over a period of 5 years. As indicated here, and in 5 different institutions. We started with 15,000, we developed a core and a mini core as well. Mini core is again about, supposed to be 10% of this, but this is not complete here, and anyway, we developed a core and mini core, which was then further subjected to develop a reference set. And then this mini core was subjected to study the genetic diversity, using 36 high variable SSR markers. And here also the message was that in some of those varieties which were released from 2006 to 2013, there's a loss of genetic diversity during this period, which was very clear message. It's already published again in I think PLOS One. So 11-13, when we started with, we did a characterization with 30 traits, and we evaluated them in 5 different institutions together, for resistance to biotic and abiotic stresses. And this program was sponsored or funded by Department of Biotechnology, Government of India. So since we got inspired, we were encouraged. In fact, next year, then we took up an effort of characterizing the entire germplasm of chickpea. Chickpea, I'm happy Dr. Chaturvedi, who was a partner in this work, in fact, already highlighted the importance with regard to sustainability in general of pulses, but chickpea is our major crop. And fortunately, we are now able to see increase in production as well, total production particularly, and also productivity in India of some of these crops. So we took a challenge again here at a single site of MPKV, Mahatma Phule Krishi Vishwavidyalaya in Rahuri, and with these 18,000 accessions, though we have about 14.5, but we took all other accessions for which we had no idea. So total a set of 18,000 accessions were actually speaking, phenotype at a single site. And these 18,000 accessions came from India, for example, 43% of it, and 32% of the germplasm was exotic from 41 countries and unknown, which is quite common, actually speaking. When you go to a gene bank, you don't know where it must have been collected from, but that material is important, therefore it always is conserved, and we can find value. And now, of course, through genomics, you can even know what that material is. So we took this challenge. Again, we developed a core set here, of about 1,200 accessions. And then finally, we also contributed with ICRISAT, with Dr. Rajiv Oshne in whole genome sequencing, which I'll talk to towards the end, and the resequencing of chickpea. And not only this was a program which we took, as a scientist, right, without any extra funding, except coming for wheat from a particular program called NICRA, but we were managing it somehow with partnership I mentioned before, for example, for pulses with Dr. N.P. Singh, Dr. Trivedi and others, and also the wheat with all wheat breeders actually speaking of the country, from the various institutions. And then we developed, actually, I was happy to coordinate this program, and I conceptualized it with Dr. Ayappan that time. He agreed and he funded this program. We call it as a CRP on agrobiodiversity, Consortium Research Project on agrobiodiversity. Agrobiodiversity, not only crops, also microbes and fish and animal species included. But this is, I'm just showing you again for chickpea. For several of these traits, then reference sets were developed by growing them in different locations, 2,300 accessions. And we've identified novel sources for resistance to Estocaita blight in case of chickpea. And of course, I was very happy, as you would have seen in the previous slide, that Dr. Swaminathan visited us in 2013 after a very long time. And quietly he went away, of course, as he normally does, back to Chennai. And then he wrote me this email after reaching, on this particular date with this message. And I am very excited and happy to share with you that I was happy to visit, of course, NBPGR again after some time. I'm particularly happy that you're converting NBPGR as a functional conservation center. Because, so far the gene banks, were only like conserving the material, which was very, very important, no doubt. But after having done that for centuries, even in India for the last about 50 years or so, we have to now start talking about utilizing this material. And that's the reason is, I said before, decreased crop diversity. So therefore he gave the word as functional, conservation center, which I think NBPGR is now looking like, since we have the material which we know what useful it is for. So we didn't stop there. And then we jumped on to the utilization of some of those crop wild relatives or wild species, particularly again in pulses. This is I had an opportunity to go to ICARDA, and met their Director General and other scientists. And I introduced, that time in NBPGR about 560 accessions of different species of lentil, and their origin is from different countries, which is a very useful resource. And then we evaluated it, here in India, in 2 different locations, actually in Delhi and Sangla for 3 consecutive years, 2011, 12, 13, 14, you see here. And we identified some of those material which is having all those important agronomic traits. And of course, this is showing again the origin of that material, which we did not have before in India. So we utilized it actually for broadening the genetic base. You would have already seen, not only in case of chickpea, but also in lentil and other crops, it is very narrow genetic base. So with the purpose of broadening the genetic base, these interspecific hybrids were made, material was generated, and these traits were developed, of different, kind of agronomically useful traits were developed, as shown here, rest resistance or some of the yield components. And finally, I'm happy that out of this all effort, in 2019, 2 varieties have been released. Dr. Mohar Singh is the one who has been actually leading this effort now, and he has been able to get these 2 varieties released, of course, by State Variety Release Committee. And these are the 2 varieties with an average yield of 10 quintals per hectare. Here, this one, 11 quintals per hectare against the Czech, which is up to 8 quintals per hectare under rain-fed conditions. So there is certainly a superiority in case of agronomic performance or yield of these 2 varieties, which are in case of lentil. Not only lentil, we introduced also chickpea, about 150 accessions, as these different wild species from different countries. We again characterize them, for the trait of interest, different species, which were either from primary, secondary gene pool. And then we again made some, to broadening the genetic base, interspecific hybrids, for different traits were developed. And then this material is undergoing now, in fact, trials, multi-location evaluation trials, of these derivatives is going on different places, like in, Palampur or in Bhopal, or this is in, Scouts, Jammu. And this is the material, which has been developed from, different. So far, reticulatum was the one, but we have now 2 other species which have gone into it. I'm sure Dr. Chaturvedi and Dr. Mohar Singh probably will know more about it. What's the final fate of it? So in fact, for helping all others, actually NBPGR is a service institution, as you all know. We try to do research, which I was at times criticized, but nevertheless, I think once we have the material, and if you're a scientist, you can sit idle on it. You have to utilize it. So we did all that actually. And finally, we also were fortunate that we were able to collate all that effort, the information on wild relatives of cultivated plants in India, and this, forward, I was very happy to be written by Nigel Maxted, who's a world authority, on crop wild relatives. This is about 750-page document. It is out of stock, I was told, but any one of you interested can write to ICR, for getting a copy. It has got not only information, in fact, which wild species is good for which particular agronomic trait is all been listed in this particular volume. Finally, as I said before, that we were happy to start with collaboration. Rajiv Oshne in 2005 came to ICRISAT, you all know, from Germany. He did an excellent job in India, while in ICRISAT, as you all know, and internationally, he's the only one scientist, I would say, who has been able to take the genomics of all these crops head on. No one else could ever imagine. And this is what actually you need, that kind of a passion to do, at the international level, whatever the best you can. To start with, actually, it was pigeon pea, but I came into picture when we, sequenced chickpea, with him. And then finally, of course, ICRISAT, and of course, many of his international collaborators included, all these pulse crops, in addition to many other crops, like, in case of millets as well, he was instrumental in whole genome sequencing and generating a reference genome. But it was not only a reference genome of a single variety, or an accession, which was important. Soon it was realized that more accessions need to be whole genome sequenced or resequenced. And therefore, here 300 accessions of pigeon pea were resequenced or sequenced, and then here 430 accessions of chickpea. And then finally, the genome-wide association studies were carried out, and genes were identified for important traits based to flowering, harvest index, or yield under drought and heat stress. And again, not only
Yes, sir. Because in
Handful of
continuous
those
after that
sources.
class,
Can you please
Corona
put off
facility
the mic?
under
Can you
continue.
please somebody mute the mic?
Sometime your institute, sir. Sometime
Can you
your
please
institute
mute
is shut
yourself?
down.
Whoever is
Sometime my
speaking is
institute
disturbing me.
because sometime no,
Abhishek,
even in
Abhishek,
our university many more
Abhishek, switch off your mic. Abhishek.
So if I continue, not only on those about hundreds of accessions, and this is the largest plant genome sequencing project undertook again by Dr. Raju Varshney and his group at national international level. I'm sure Dr. Chaturvedi and Dr. N.P. Singh and others, are included in this effort, wherein they sequenced about 3,366 genome and developed a pan genome. I'll just talk to you a little bit about it. And we were fortunate that we were invited by Trends in Plant Science to write a spotlight on it. And the clear message we gave that such a large genome sequencing project, has highlighted that how this breakthrough of research now can fundamentally change breeding practices of chickpea, and not only chickpea, of also potentially other crops. And this was a breakthrough, I would say. It's not easy again, but it was done. And more than rice is the only one example otherwise, which I think Dr. Anil Kumar mentioned, where 3,000 accessions of rice by IRRI and other partners have been sequenced. But this is the largest, beyond 3,000 gone into a crop like important crop for India, chickpea. So finally, out of these 3,000 odd genome sequences, a pan genome was developed of 592 megabase, which included a reference genome, of course, earlier developed an additional megabases from a desi genome and also from a wild species reticulatum, which is possible with size of erythema. And important is that finally they found some new 1,600 genes identified from this pan genome. And out of these 1,582 potentially novel genes, maybe some were already known, but this was a large chunk of it were novel. And more importantly, that they were able to increase the number, as the number of individuals sequenced increased, the number of dispensable genes increased. And actually, this is the number. These are the genes, dispensable genes, which are important for providing resistance or tolerance to various biotic and abiotic stresses. And this is what is the advantage of doing this, whole genome sequencing. So finally, these studies have provided a kind of a platform, which Dr. Anil Kumar also mentioned. We call it a genomic assisted breeding approaches. Earlier, when we talk of GAB alone, genomic assisted breeding, it was like marker assisted selection, marker assisted backcross breeding, marker assisted recurrent selection. But now there's a possibility based on the genomic selection and genomic information available. You talk of haplotype based breeding, or genomic selection, or optimal contribution selection. Optimal contribution selection means here that you have got, including your pan genome, several of those land races and even wild species. And they would have some of those deleterious genes as well, or alleles, which are not supposed to be carried forward into a variety. So there is a possibility you can have an optimal contribution, only of those favorable genes and alleles, and you can discard many of those, deleterious alleles and genes, when you start up developing varieties based on the genomic assisted breeding platform. And of course, through this is nothing but genome editing. Again, through genome editing, you can do what otherwise is possible through genomic, but here you can do it in a much more precise and accurate and targeted manner. And of course, genome editing, I don't want to go into the details of it, actually speaking, maybe some other day, but here only the message which I want to give you, this is, as Dr. Anil Kumar also mentioned, out of the two, or Dr. Chaturvedi also mentioned, the two of them who got the Nobel Prize, she's actually Jennifer Doudna, from USA, and she was making a presentation somewhere, and I happened to, get hold of that slide. So a friend of mine, which she made a presentation on October 5, 2021. This is her own slide she was presenting with regard to agriculture. And her message is that CRISPR-based solutions will allow farmers to adapt to a changing climate and sequester more carbon. Actually, she was emphasizing more on this. Sequester more carbon while preserving prized regional varieties, which prized regional varieties are nothing but we're talking of those land resources or farmers' varieties. And there is an incredibly important role that these genome editing technologies tool can now play, in providing kind of livelihood security or protection of some of those smallholder farmers. And of course, this is very important, and I'm sure the whole world is now engaged in it. I don't have to go into the details of it, but only thing I want to mention here again to the younger scientists and colleagues, while it looks very fascinating that, okay, I would like to have a project on genome editing, or would like to genome edit a particular crop, but important is that you need to have a highly efficient regeneration and transformation protocol is very, very important. Unfortunately, we don't have one at the moment in our country. Of some of the pulse crops, particularly, I want to give emphasis on, again, beyond wheat and rice or other cereal crops. Of course, millets are again important. But pulses, for sustainable production point of view, thanks to Dr. Chaturvedi to bring this point home, it's very important because they only have the capacity of, biological nitrogen fixation. So therefore, in India, there's a potential that we are the largest producer of pulse crops, and consumers also of this pulse crop, produce. So we should be taking this head on. And this one paper I came across, wherein they have developed for the first time, they claim in cowpea such a protocol, and therefore it will be utilized for genome editing system. But what is important is we need this in chickpea. Of course, I think it is there in IIPR. I'm sure Dr. Chaturvedi knows about it. Also in pigeon pea, lentil, urd bean, mung bean, moat bean, rice bean, and some of these underutilized beans are very, very important nutritional point of view to tackle the issue of malnutrition. And more importantly, we can even try to now domesticate these wild species and accelerate that process through the process of genome editing, as it has been already demonstrated in case of tomato. Wonderfully, only by about multiplex editing some of 5 to 6 gene or targeted regions in certain genes with regard to architecture, phenotype, etc. Tomato has already been demonstrated to have in this accelerated process of domestication through the process of genome editing. So this is a challenge, however, in our country that we should try to take up much work. And I hope, Dr. Chaturvedi, Dr. Arvind Kumar as the Honorable Vice Chancellor, Dr. Anil Kumar, all together can probably take this lead in this university and elsewhere as well in a collaborative mode. So finally, friends, I would like to conclude that an accelerated breeding system is important for developing these crops, which are climate resilient. But what it requires is a good understanding of the molecular and genetic basis of some of those traits which are related or, with adaptation to climate change. There could be many, could be resistance or tolerance to various biotic stresses, etc., or new emerging pests. But we need to understand the molecular and genetic basis, and therefore, and also for this, what you require is the genetic resources, which have been collected. As I give you a good picture about it, I thought, in case of, India and elsewhere, they're there. These materials are all available. And these have been collected from regions with varied climatic conditions and subjected to different climatic regimes over a long period of time. So they carry those alleles, actually speaking, which we have been missing out on them. Therefore, pangenomics become very, very important. We must have then now taken, we must take these exercises more into other crops as well. So finally, again, I come back to Gene Bank. And Gene Bank, which you all know, is a collection largely comprising natural populations, and they have assembled over several decades, provide a valuable resource for crop improvement through genomics and genome engineering approaches. So, and not only, when you talk of genomics and genome engineering approaches, you need kind of information on the sequences from those genomic regions, which are controlling particular traits. And therefore, what you need is a biodigital gene bank. So from Functional Conservation Center, which our Dr. Swaminathan sir mentioned about 10 years ago, I think we need to now move to biodigital gene sequencing, because there is a lot of digital sequence information which is being generated, through these thousands and thousands of accessions in different crops. So this is actually the same thing which I mentioned before, to utilize it, these are different crop breeding strategies or pangenomic analysis, or functional discovery of various genes, and finally, all that information to be available in a gene bank, which we call it as a biodigital gene bank. And that's what, in fact, is the image from the Trends in Plant Science paper, spotlight, which we put together. So with this, I thank you all for patient listening. And before I thank you all, of course, I have a pleasant duty to acknowledge, some of my colleagues, of course, all scientists of NBPGR, who gave me this opportunity and strength to do the work which I could do. But particularly in case of pulses, I have some information gathered today from Dr. Gayacharan, Dr. Kuldeep Tripathi, Dr. Mohar Singh, and of course, Professor Rajiv Varshney shared some of his slides, and I'm very thankful to him. And I'm thankful to all of you for your patient listening. I'll be happy, if time permits, to take a few questions as well. So thank you again, Professor Anil Kumar, for this opportunity, and all of you at the Jhansi Central Agriculture University.
Thank you so much, sir, very much. For sharing your views on genetic diversity, genomics, and genome engineering for crop improvement. Nice presentation, sir. Your slides are self-explanatory. You emphasizes on research as well as government priorities to meet sustainable development goals by 2030, and also utilization of crop diversity in a sustainable way, keeping in mind the past lessons along with way forward. Your ideas and experience on plant genetic resources along with plant biotechnology will definitely help all of us to move forward in order to meet various sustainable goals. Thank you, sir.
I have just two small comments, sir.
Yes.
Can I?
Please.
Sir, pranam.
Pranam.
So the first thing that really always I appreciate the efforts made by you under your leadership to characterize chickpea and wheat germplasm. It was not a small thing to initiate and complete it. That's
Thank you.
Sir. Secondly, you said that less than 1%, hardly 2 or 3 germplasm material has been really utilized for development of varieties. Those varieties could see the day of light of the day. So what really you feel? Whether the germplasm is not worth, that was not worth, or it has not been characterized, or the value had not been added to those germplasms. That's all.
Actually, this is again a wonderful question and clarification. I think will be helpful to others as well to know. I would say that we have not been able to characterize, evaluate, and only if at all we were able to do only handful of the total collection. For example, in case of wheat, I talked about 22,000 sessions, though there are in fact internationally, as you know, even many more in number. But when I was there in NBPGR, they would be doing 1,000, 2,000, and one day on a field experiment where all breeders, mostly of wheat, were present, I asked them, have you seen any kind of genetic variability? What is the largest amount of genetic variability you have witnessed on a single site, whether 1,000, 2,000, 5,000, or 10,000? I mean, this was mind-boggling. I said, here, I have got before you on a single site more than 20,000 accessions genetic variability. So the point was, while the efforts were on, but they were not on entire collection, number one. And number two, I don't blame, I said before, the breeders, because they didn't have enough information on the germplasm to utilize. Pre-breeding, no paper will give you, you will not have any kind of variety developed out of this effort. So unfortunately, our policy is also at fault, because we go by how many number of varieties any breeder has developed. We don't go by any pre-breeding effort which has been made and somebody has developed those, developed those traits, and you have really prepared that material for utilization by plant breeders. You will not get any marks. Even you will not get very high quality papers. I mean, I was also subjected to this kind of discussion with my colleagues that if you do this characterization evaluation, will we get good papers? And I was fortunate that we have published all except 1 or 2 which are in the process in the last about 10 years. We must have got, in fact, and some of the material we have generated, in chickpea, we shared it with Professor Akhilesh Tyagi, at NIPGR. He was director at that time there. And Dr. Savrut Parida, who will be making this presentation today, probably he'll talk about it. We gave this material readily to them, to utilize it at the genomics level. So therefore, in fact, it is team effort was lacking, I would say this. And we are always in a hurry to develop a variety with whatever the genetic stock and variability we have with us. And so that we then get only, the promotions which are important and the policy are such. But otherwise, a long planned strategy was missing, and it's still missing. I would say this, really speaking, in majority of the crops, we must have, I talk about it mostly as PGR to product, plant genetic resources to product development in terms of a variety of a particular crop. We must have a thorough continuum and a long-term strategy on a mission board, particularly many of these crops. And that's what actually Rajiv Washington took the benefit of the gene bank material in ICRISAT, or in Africa or in other countries. Therefore he could develop this pan genome, with such a large number of accessions in case of, chickpea. And of course, he's doing other crops as well. So the gene bank material need to be highlighted, needs to be utilized, need to be valued, and need to be respected by one and all. It's very important.
Second, just a small, you rightly said about that genome editing, Prakash and all.
Yes.
So what do you see that when we are going to utilize this particular technology, we can make corrections in the ongoing available material, including by relatives. So really, is there need to continue with the large collection of, let's say, 19,000 germplasm accessories of chickpea at NBPGR, another 18,000 at we said, 8,000, 9,000 at Icarda? Is that required? Or we can meet out our demand through genome editing?
You know what?
How you see that germplasm will move?
No, you're right. It's a good question. But again, I would like to mention here that, my dear Dr. Chaturvedi, to know which gene you are going to edit and what are we going to edit finally, what we are going to mimic, the naturally occurring variation. We want to bring it into our high yielding popular varieties. And how do we know what is that kind of naturally occurring variation or that particular allelic difference we have it? That we only get it from those entire germplasm collections we have. So that you have to always maintain. What we need to do is we have to create those pan-genomes. We have to have that kind of a relationship of those genomic regions and molecular bases I mentioned before. Then only you will know what gene to be added, to be deleted, to be gain of function or loss of function, or what kind of bases you need to mutate in a promoter sequence or an exon sequence. That information you will only get it from these naturally occurring land races, farmers varieties, kind of genetic variation they have. Okay, and another thing with regard to wild species I mentioned, we need to maintain those and even collect more of wild species, globally speaking, because then, while we all know they have deleterious alleles, but then if you have got a genome editing protocol available, then you can delete the function of those deleterious alleles, and as it has been done in case of tomato, and you can bring we can do that accelerated domestication. So this physical seeds conservation in any of the National Gene Bank or internationally is of paramount importance. It cannot be undermined, even what technology might come across further in the future as well for crop improvement. It's very important.
Thank you, sir. Because many students are asking, so I thought that it would be good if you clarify.
Sure. No, thank you. I
thanks,
appreciate
thanks for excellent talk. And always I enjoy your talk. You are excellent speaker.
Thank you.
Very good knowledge you have.
Thank you.
Thank you, sir.
And you talked about genome editing in India. By the way, we're already working. In fact, speaker, Dr. Ashwini Parikh's institute has genome edited. I see. We have genome edited rice. We have other genome edited crops.
Many crops are coming.
People working already in India.
Many crops are. Thank you, sir. Thank you.
Pleasure.
Thank you very much for giving your nice presentation. Indeed, you have cited very good example of tomato, where this pimplicifolium is converted into esculentum, and this is 6 SNP variation, which has been engineered to have now the modern cultivar. And since it is happening since ancient time, domestication process is going on. So how do you think that these allelic variations, which is occurring during domestication process and modern genomic selection, how will you say that really they are favoring whenever you are doing genome editing technique, whether they are really favored or naturally occurring process, they are really favored, so that you will get a desired trait? So this is just, I want to ask whether really genomic selection is going to give more clues so that you can do the targeted breeding program.
You know what, again, Dr. Anil Kumar, that which allele is deleterious or which allele is useful, though they are there, in all these wild species, we know that, in large numbers. But at the same time, you will only get this information once you have a genomic sequence available and you have been able to compare it with the reference genome, or you have been able to develop a pan genome. Because then only you will be able to, and through the process of genome editing, then you have to identify the ones, comparatively, you can then identify what are the genes. And through genome editing, yes, it is possible, particularly through genomic selection, of course, and also through genome editing. Now, there are cases wherein 10 to 12 genes together can be genome edited at a single go, So therefore, this is a possibility, while haplotype breeding will be possibility through kind of genomics approaches, but genome edited breeding is very much possible. But only thing is you have to generate the large genome data, and you have to do all those genome-wide association studies using those panels, including land races and wild species. to identify what are those alleles which are deleterious or which are favorable, and you can then slowly bring them together in a single genetic background, through genomic selection or also through genome editing together. Two parallel approach it can be, but you need to generate a lot of information data on genomics first before you embark upon such a study.
Because, sir, until unless you don't have idea about the important alleles or the SNP variation, which is contributing for particular agriculturally important trait, like in the rice, grain size that is being only by single mutation in the ubiquitin ligase. If this single mutation can increase the size of the rice grain. So if such thing is happening, so
Dr. Anil Kumar, that information we are talking about rice came from genome-wide accession mapping studies of thousands of accessions of rice. They developed a panel and they then genotyped it and phenotyped it 14 different agronomic traits, and therefore they could have this kind of a association of the genomic region with the phenotypic trait of agronomic importance, and they could identify that single gene. And finally, they could, through the transgenic approach, they could show, yes, it works very well.
Thank you, sir. Thank you very much.
Another advantage of the genome editing technology would be, while you can always cross wild species, some of them are incompatible with when you talk of sexual hybridization with the cultivated species. And in case of rice, there is one example in Niri which has taken 20 to 30 years. From wild species integration of a gene into cultivated tag, they have not been able to do it. But if we have that information and knowledge through genome editing, you can do it in less than 5 years.
Thank you very much, sir. Thank you very much for your enlightening talk and also solving the queries raised by many of the participants. Thank you, sir.
Yeah, I wish the participants had come directly also to talk to us later, maybe, or they're waiting for Dr. Ashwini Parikh's presentation, which is enlightening, and I'm further looking forward to it myself. Thank you very much for inviting me once again.
Thank you, sir. Now I request Dr. Nishant Bhanu for a brief introduction of our next speaker and guest of honor for this program.
Thank you, sir. I'm glad to introduce guest of honor for this event, Dr. Ashwini Parikh, Executive Director of NAVI, who is a prominent plant biologist and educator, noted chiefly for his contribution in plant molecular biology and biotechnology. Dr. Parikh received doctoral research PhD in plant molecular biology from the University of Delhi and is currently working as Professor of Plant Molecular Biology and Biotechnology at the School of Life Sciences, Jawaharlal Nehru University, New Delhi, and Adjunct Professor at the University of Western Australia, Perth, Australia. Dr. Parikh received the prestigious postdoctoral fellowship by the Rockefeller Foundation, New York, and worked at University of North Carolina, USA. Later, Dr. Parikh was awarded Boycott Fellowship to work at the University of Illinois at Urbana-Champaign, USA. He was also awarded the INSA Royal Visiting Scientist Fellowship to work at University of Cambridge, UK. He has also been conferred upon the DBT Overseas Visiting Fellowship to work at the University of California, Davis. Professor Parikh is a recipient of several honors, including the Visitors Award for Technology Development from the President of India for developing stress-tolerant rice of the next generation that has potential to enhance the income of rice farmers. Recently, he has been awarded Tata Innovation Award by Department of Biotechnology, Government of India. He has been elected Fellow of the National Academy of Sciences, India, and National Academy of Agricultural Sciences. He is a recipient of various prestigious awards such as Chinmay Gold Medal Award by Indian Society of Plant Physiology for contribution in the area of plant science. He has keen interest in understanding the physiological and molecular adaptations in zero halophytic plants and development of transgenic rice plants with enhanced tolerance towards multiple abiotic stresses, including salinity and drought. He has authored and published more than 150 research papers in the reputed research journals and several book chapters and edited books also. It is our privilege to have you with us today, sir. Most welcome, sir. Sir, I invite you for your lecture.
A very good afternoon to all of you. I hope I'm audible to all the participants. Thank you very much, Honorable Vice Chancellor of Rani Lakshmi Bai Central Agricultural University. I was quite impressed, by the way, to have a look at the video which was shown in the beginning. I think within a few years of span of time, this university is taking good shape, and I'm very pleased to see that under the leadership of Dr. Chaturvedi and Dr. Anil Kumar, I think I can see exciting things happening for not only the students of the university, but using this innovative virtual platform, which is quite common now. I think the information is big spread to all the students and those who are interested. Thank you very much. I am very pleased and honored to be a participant in this national webinar. And believe it or not, it is very difficult to speak after Dr. K.C. Bansal, who has been a wonderful researcher, wonderful speaker, and a wonderful human being, no doubt about it. So he has set the bar very high. But nonetheless, since he's sitting over there, I will have to perform my duties, and I'll try to do as smooth as possible. So the speakers have already, give the information about the diversity, the importance of diversity. Germplasm, Dr. K.C. Bansal very beautifully summarized, and he explained with the help of his effort and NDPGR that such kind of, information, such kind of knowledge, such kind of germplasm conservation is extremely important. So my job over here in the next 30 minutes or so will be to, take you deep dive into one direction and show that how this allele mining with respect to, handling, understanding how nature is tolerating different kind of stresses. That's what I'm going to talk about. I'm going to talk about the diversity. I'm going to talk about how the wild relatives are actually important and how molecular biology tools can be actually used to do something, to make something very useful in a changing climatic condition. Of course, I have several hats over here, but I would like to restrict my today's talk to my first hat. Let me use the pointer over here. I would restrict my today's presentation to the first affiliation, which is Jawaharlal Nehru University, primarily meaning that I'll be presenting the work which we have carried out at our School of Life Sciences, Jawaharlal Nehru University, and I would take opportunity to present the work which is being done at other excellent work being done at RLBCAU and other places in some other presentation. Please allow me to move to the next slide. Just a brief introduction to see that why we need to develop plants which are resilient to stresses. Dr. Bansal has said enough, but just to revive what I'm saying, that the world population is increasing every second, and if you go to the clock which are available virtually on the screen, the clock goes like tick 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 tick, and it is very scary to actually see that we are increasing number at such a high speed. Once you look at the clock, you will be very surprised. And there is another clock available at many of the websites, which also tells you that it also goes tick, 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 but it goes in reverse direction. And that, my dear friend, is the number of the land which is available in hectares. And this clock goes like in the reverse direction. So on one hand, we have the world population, which is increasing every second. And on the other hand, we have the land, which is being used for agriculture. There's a kind of a double-edged sword we are talking about. And on top of that, we also have stresses. I don't have to emphasize the importance of stresses such as salinity, drought, insect, viruses, nematode, so on and so forth. And these all together are actually making us think about it, that will we have enough food available to be able to feed 9 billion, 10 billion? And my dear friend, if I tell you the reality in India, the population is increasing at such alarming rate that I'm told that we are going to outcompete China, at least in one thing, and that is the number of population. And the graph on the left side seems to be very interesting, where it is clearly shown that the population of many of the states, which includes UP, Haryana, Punjab, Rajasthan, Bihar, Maharashtra, is actually equivalent to the number of population in many of the countries such as Mexico, Germany, Vietnam. You must have seen this slide at some places as well. But I just wanted to draw your attention that it is a big challenge for us to ensure the food security in light of this number, because we don't have to feed 27 states, we have to feed 27 countries within our country. That's the challenge I'm talking about. That's the challenge I'm talking about, the food security. Many of us who are feeding on rice and wheat as a natural source of carbohydrate do not even get access to pulses. And it has been very well emphasized the importance of pulses, because the food has to be nutritionally balanced. On one hand, we are talking about nutrition security along with the food security, and we need to understand how we can actually use the tools of molecular biology or contemporary tools of genomics and breeding to make the food which is not only available in plenty, but whatever is available is nutritionally balanced. So these are the two challenges which I'm talking about. On top of this, we also realize that a farmer does not want his son or daughter to continue farming as a profession. That's the societal problem. And based on the last two census, I'm told that 4,432 farmers are discontinuing farming as a profession every day in our country. I would repeat, 4,432 farmers are discontinuing farming as a profession in our country. Who's going to continue farming if it is not going to be promising in terms of return of investment? That's another problem. And the land holdings, all of you know, are becoming smaller and smaller every day. So these are some of the issues as a scientist, not only as a student of agriculture or biology, we need to understand. But coming back to today's topic, which is related to allele mining, we are working on salinity and drought because these two stresses are actually very, very important as far as the Indian agriculture is concerned. And to support my statement, I would like to draw your attention to the cover page story, which was published on Nature, where they did some analysis with the help of satellite images. And the circle which I'm showing over here, which includes states such as Haryana, Punjab, Rajasthan, where I'm sitting at the moment, Delhi, which is also considered as a food basket. And they have been shown that within the year of 6 years, from 2002 to 2008, roughly more than 100 cubic kilometers of groundwater has been extracted. The reason being that the monsoons are becoming unpredictable, farmers are extracting more and more water, which is not only making this land, the food basket of the country, as a thirsty land, but also because of the irrigation practices, we are increasing salinity level. The meaning thereby that salinity and drought remains a serious threat to agriculture as far as India is concerned. I'm going to talk about one of the important crops. You listened about the chickpea. I'm going to talk about another crop, which is rice. For obvious reason, rice is the food for richest, rice is food for the poorest, and there is a lot of genetic diversity which is available within this genotype. That gives the molecular biologists a wonderful tool to handle. I'm going to talk about the diversity and the salinity and the drought, how we can do it. So soil salinity is an important issue. If it goes, the electrical conductivity goes above 4 decisiemens per meter, we call it saline, and it causes several effects. It causes iron toxicity because sodium is very, very toxic. It removes the hydrogen from the proteins and the protein becomes denatured. It also alters the potassium-sodium ratios. Potassium is very beneficial for the cell for various reasons, stromatal opening, and sodium is toxic, so the ratio is disturbed, and also it actually disturbs the nutrition balance as well. Meaning thereby that salinity affects the normal physiology, metabolism, and that results in the growth inhibition, which is very, very important because ultimately leads to decline in crop yield. So those who are interested to know more about it, there is a video available which was made by New York University, Cornell University, Pursuing a Dream. They can just Google around, and the details of work we are doing will be available wonderfully on this video, Pursuing a Dream. Since this workshop is specifically about allele mining, I thought of picking up some work which is relevant to this webinar. And I'm very happy to say that we have enough diversity as far as rice is concerned. And when it comes to salinity in rice, the obvious thing which comes to our question mind is that what do we do in the coastal areas? Because coastal areas have high salinity. And when you look around, you find that, yes, the coastal farmers are very, very smart because they have traditionally been able to grow a rice paddy, which they call pokali. And this pokali is good enough, as you can see over here. It can be grown even in sea water. Practically, you can see what I'm showing over here in the image. And it gives you beautiful productivity, which is good enough for the farmer to survive. But the problem is that the yield is not so high because yield is compromised because of the tall nature of the rice and also the salinity, which, as I said, results in it. But nonetheless, this is a wonderful germplasm to study salinity because it still gives you productivity under natural saline conditions I'm talking about. So we thought of doing allele mining from the pokali rice, which is wonderful rice, as you can see over here. And we have done a lot of work. Today's work, I will be restricting to only 2 examples. We have at least patented 2 dozen gene from this particular plant, pokali. I will restrict my study. So the work was, appreciated, and it was a newspaper item in many stories of pokali and pokali rice going places. This is just to show that, when you work on your local germplasm, as we are talking about conserving the local germplasm, Dr. Bansal also mentioned, you are actually appreciated by one and all. So I think there is a need to go more deeper into these kind of work, which is going to not only bring in the values, but also some substance into that. So we thought of, using a very powerful approach, and the approach I'm going to describe for the benefit of students, since I know many of the students are attending this webinar, is called contrasting genotype. Dr. Anil Kumar would agree with me that this is one of the very powerful approach in the sense that you have a rice variety which is salt sensitive, which is IR64, high yielding, but very, very sensitive to salinity. And on the other hand, you have a natural selection wild rice, which is highly tolerant, but giving very poor yield. And these kind of contrasting nature is something a gold mine for a molecular biologist like me. You can actually do deep dive into the molecular mechanism using these two contrasting genotypes, and that's exactly what we did. We first characterized this IR64 and Pokali. On the left side, you can see this is IR64 control. This is IR64 stress. Again, Pokali control, Pokali stress. These are the seedlings, just to show you that even at the seedling stage. By the way, I would like to tell for the benefit of students that seedling stage, we restricted our work to because seedling stage is the most sensitive stage as far as rice plant is concerned. So we wanted to work at the stage which is actually relevant as far as sensitivity is concerned. So no matter what you talk about after the relative water content, root length, shoot length after 24 hours, 48 hours, you can clearly see that Pokali is very, very happy as compared to what you see in IR64, meaning thereby that the system is actually within the condition which we are using, 24 hours, 48 hours, 72 hours. These conditions are able to show the contrast. And this is exactly what we needed. And once we had established this contrast at the seeding level, then the next point was to, go deeper into it and do analysis. When we did the analysis for atomic absorption spectroscopy or X-ray diffraction, what we found is that Pokali, as you can see, this is EDXRF instrument. After 24 hours, you can see IR64 start accumulating NaCl, sodium you're talking about, while in Pokali, you hardly see any sodium buildup. And after 48 hours, you see high amount. And after 72 hours, you see that high amount of sodium is accumulating beside many others shown over here. Sodium is the one which is highly toxic. And you can see the difference between IR64 and Pokali as far as sodium availability is concerned, meaning thereby that the plant are not accumulating sodium or accumulating less sodium in Pokali and more sodium in IR64, meaning thereby that even at the system which we are standardizing, we have good evidence that yes, sodium is entering and it is causing toxicity in the seedling. These are other parameters which we analyze: the decrease in sodium potassium ratio, electrolyte leakage, increase in protein content, clearly establishing altogether that yes, seedling state is very, very important. And within the conditions of 24 hours, 48 hours, 72 hours, you have differences in many of the aspects, physiological aspect, biochemical aspect, like, you're talking about membrane damage in the form of melanoaldehyde. You're talking about protein accumulation, which is a osmoprotectant. So no matter what parameter do you take, there is a contrast established even at the seedling stage at 24, 48, and 72 hours. Going deeper into that, we established with the help of corona dye, which is specifically, this is not coronavirus, by the way, this is the corona dye I'm talking about. Coincidentally, the name is very, very, I would say, scary at the moment to talk about, but this corona dye is binding specifically to sodium. As you can see, the IR64, it has more sodium accumulated as compared to Pokali. And when you do the tunnel assay, which talks about the DNA damage, it is much, much lesser. So what I'm trying to establish over here is that Pokali shows all parameters which are showing strong phenotype as compared to IR64 when subjected to NaCl. And Dr. Anil Kumar will be very happy to see this is the image which we generated by PAM. Photo amplitude modulation, pulse amplitude modulation, and the blue color indicate that yes, the plants are still photosynthesizing when they are not happy as far as photosynthesis. This is actually indicating the FPFM ratio, and you can see very clearly the Pokali seedlings are very, very happy till 72 hours of salinity. You can still see some color, but in case of IR64, they start dying after 72 hours, 48 hours. And though, no matter again what parameter do you take, these are OJIP curve, which talks about photosystem 2 and energy trapping, electron transport. We have done detailed analysis. I will skip the detail, but those students who are interested, they can just log on to the PubMed and get the details on that. To conclude this first part of my talk, I would like to draw your attention towards it. IR64 is sensitive because it has a high buildup of H2O2 hydrogen peroxide. It has ion imbalance. Vacuole has more sodium, proline less accumulation, degradation of photosynthesis, limited carboxylation, basically meaning dysfunction chloroplast, which results in decline in crop yield. Finally, we're talking about. On the other hand, the Pokali, which is tolerant, it has all the parameters, but in a favorable manner. You talk about biochemistry, you talk about molecular biology, you talk about physiology, everything is much more stronger as compared to IR64, as we can see. Having established that, yes, we have the contrasting system, then we wanted to use the omics platform, which many of you are aware of it. When it comes to an omics platform, we do proteomics for analysis of all the protein which are up or down regulated, giving it an idea about translation process. We do ionomics, as I've already shown one part of it, where we see how many ions are accumulated, which ratio and differentially accumulated. Then we go back and trace the generation of this proteome by using transcriptomic, where we do real-time PCR, we do NGS sequencing, we do northern blotting, reverse northern blotting, and a lot of things we do. When it comes to proteomics, we do two-dimensional gel reductor fluorescence, we do eye track, and so on and so forth. When it comes to metabolomics, we analyze carbohydrate, proteins, organic acid, and so on and so forth, using GCMS analysis and UPLC analysis. So we have used a combination of instrument, combination of these technologies to go, deeper into the differences, why IR64 is sensitive and why Pokali is tolerant. For the benefit of student, I would like to draw an analogy. What I'm talking about is the kind of systems biology approach, where in a room, it has say 4 or 5 windows, and when you peep from one window and try to see what is there inside, and then you peep from another window, then you peep from another window. So this gives you a kind of, three-dimensional view that there is a one chair, one table available in the room. So these kind of, tools allow you to look from different windows what is actually happening in a cell at the cellular level, organ level, as well as the whole plant level. And I will skip the details of it, but give you one important aspect of transcriptomics, which will help me in building up the next story. We use different kind of methods for transcriptome analysis. Another method which I still believe is much better than what we have the modern tool is a subtractive cDNA library. Very old tool, but very, very powerful tool. Those who have experienced subtractive cDNA library, they will agree with me. What you do in this case that you take out mRNA, you actually mix up the two mRNA, convert one into cDNA, there is a hybridization, and those mRNA, those sequencing, those molecules which are differentially accumulated, then you bring them out and clone them in a library. Then you sequence the library, and this is what is subtractive cDNA library we are talking about. We actually made several subtractive cDNA libraries between IR64 and Pokali, and I would like to draw your attention that one of the unique things which we did at seedling level, that we actually made a library by pooling in the sample of the seedling which were subjected to stress for just 10 minutes. 10 minutes, because we wanted to see that within 10 minutes, what happens to the signaling gene? What are the primary transcripts which are actually upregulated, downregulated? Another library we made at 20 minutes of stress, 30 minutes of stress. And of course, we did 48 hours of stress, 72 hours, 96 hours, because these kind of time kinetics allows you to actually peep in a time-dependent manner what actually is going on in the cell as far as the transcriptome regulation is concerned. Just one representative library I'm showing over here. When we sequence the gene, we annotate those genes, and for the benefit of students, what you do is that you sequence those genes and go to the PubMed, do blast analysis, and then you try to see that what is the identity of that particular gene. When we analyzed these genes, we saw that many of the genes related to protein synthesis, processing, stress-induced genes, signal transduction, DNA metabolism, protein degradation, and so on and so forth. Meaning that by that, within 10 minutes of stress, the transcripts which are contributing to these different processes, they get differentially regulated. This is quite surprising. Within 10 minutes, it happened. And what is even more surprising is that the rice genome sequence was sequenced, in the year 2002, 20 years ago. But 18% of the transcripts have no significant hit, and 20% of them are still hypothetical. But believe it, they're differentially regulated. Meaning that by that, almost half of the genome, at least 38 or 40% of the gene of the transcriptome, is yet to be understood. What these genes are, what they are coding for. That's the question. This is untapped, resources I'm talking about as far as, and when we put them on the different chromosomes of rice, we find that chromosome number 1, 2, and 3 have more ESTs. The circle represent the ESTs, which were differentially regulated. And similarly, we did proteome analysis, metabolome analysis. I will skip the details, but any of you who has interest, they can write to me at my Gmail address, ashwaniparik@gmail.com. So once we understood that, yes, there is a contrast, and we need to go for allele mining. We actually did allele mining from Pokali for almost 2 dozens of genes, which are listed over here, and most of them have been patented. So on one hand, we actually isolated this novel gene, allele rather, from Pokali, did sequence analysis, bioinformatic analysis, and also functional validation by overexpressing them or knocking down them, and try to find out if they have any role as far as stress tolerance is concerned, because they are differentially regulated. That's one route we took. And since Government of India is yet to approve the use of genetically modified crops for consumption of human beings, we actually took another route as well, where we did forward genetics, where we created mutants, subjected them to the stress, whichever mutant was performing best. This program was supported by FAO for the last 20 years. They are supporting us for this program. And we actually found a lot of good mutant. And once we have the mutant, It's a gold mine for a molecular biologist. Dr. Anil Gupta and Dr. Bansal will agree with me. Once you have IR64, which is sensitive, and a mutant of IR64, which is drought tolerant, stress tolerant, this is the gold mine. And this is exactly the route which we are following. For today's work, I will restrict, since the topic of the webinar is allele mining. I would take one or two examples from this battery of the gene, which is listed on the bottom right, and explain it to you for the benefit of student, how you can move forward from the basic science which I just presented and move into translation mode to go for commercialization. One of the gene, which is a membrane-bound histidine kinase, which we did a lot of genetic analysis, microscopic analysis, and we showed that this is a osmosensor we are talking about. And remember, could you please mute yourself? It's a request from my side. Yeah.
Kartike, please mute yourself.
Thank you very much. So this particular membrane-bound protein is actually acting as an osmosensor. Again, I will skip the details, but any of you, if you're interested, will be very happy in this. This actually is a dimer, which is a histidine kinase. So autophosphorylation takes place as the conserved histidine residue, then it is transferred to aspartate. This gets phosphorylated. Then there is the shuttle protein, which we call phosphotransfer protein, which get phosphorylated. And this protein, once it is phosphorylated, goes into the nucleus, and then it actually activates several response regulator. Many of them have a DNA binding domain, meaning thereby they're acting as a transcription factor, and then hundreds of battery of reported of gene get activated. So the kind of cascade, also osmosensing cascade also we discovered, and I'm very happy to say that this particular pathway we are actually following it deeply, and we have at least a dozen of transgenic for these RR22, 26, where knockdown has been taking place, overexpression, and this is giving us beautiful insight how the signal transduction cascade is actually moving as far as osmosensing, sensing of various hormones is concerned. This is mind-boggling actually to understand the complexity, the uniqueness between the transcription node as far as signaling for stress is concerned in rice. Nonetheless, to conclude the story, second part of my talk, that when you overexpress this particular gene, you actually get 30% higher yield than the wild type gene. And this particular product has been patented in almost a dozen countries, and we have moved into translation mode where we have joined a company. Company is now trying to commercialize this not in India, because India has not given permission to grow transgenically modified crop, but the company has found that yes, this transgenic plant which we have generated, which we call strong rice, stress tolerance rice of the next generation, is actually performing well under field conditions as well. So this is, I think, a very unique story where we learned from Pokali rice. We did basic analysis. We picked up the first protein, which is an osmosensor, and modified it. This is a root-specific protein, membrane protein, and now we have a product available. So this is a good example for the benefit of students. I would like to say that simple understanding of allele mining from wild relative has actually resulted in development of a product which is commercially going to be available very soon. And I'm very happy to say that this could fetch us award from the president himself. We were invited to the Rashtrapati Bhavan, which was a very pleasurable moment for us in the year 2018. The best technology award was given to us, and this rice has been named as stress tolerant rice of the next generation. In short, we call it acronym is STRONG. So we are trying to make rice strong. The next few minutes I will give you another example where we are taking a lead from the breeding, where we have identified, scientific community has identified a QTL on chromosome number 1, which we call sorbitol QTL. We analyze this QTL, almost 900 genes are associated with it. At least a dozen of them we have analyzed by functional genomics. And these are transcription factor and signal transduction and so on and so forth. Several of the genes we have characterized in our laboratory. But to cut the story short, one of the protein which we found has a strong homology and having a coiled structure. So this we thought, if it is having a coiled structure, it's a cytoskeletal protein. And then we looked at the literature, we found that yes, this is cytoskeletal protein, and this belongs to intermediate filament. And when it comes to intermediate filament, the plant cytoskeleton, the textbook information tells us these are microtubules, there are microfilaments, which play an important role in abiotic stress tolerance as well. But the importance or the existence of intermediate filament has not been shown in plant. So for the first time, very fundamental discovery has been done where we have reported intermediate filaments from plant, and this is widely published now. Several people have followed our work, and we are very happy to say this is the basic contribution, fundamental contribution, which we have done from our lab, which we have not shown that the intermediate filament exist in plant, but they have role for abiotic stress tolerance. Obviously, this is a cytoskeletal protein, and I'll show you how it is important. This particular intermediate filament gene is differentially regulated in IR64, the white boxes we are talking about, and the filled one in Pokali. As you can see, one under controlled conditions also, that means non-stress condition. Pokali has higher abundance of the transcript, which is shown by the black bar under non-stress condition. And of course, it is then upregulated in response to several factors, which include dehydration, high temperature, abscisic acid, heavy metal, methyl violation, meaning thereby that this kind of gene is multi-stress responsive. And since we are talking about climate change, having a gene which is multi-stress responsive is very, very important. So we did subcellular localization. I will just flash through these slides. And when we express this gene in E. coli, as you can see, these two bars on the right side are showing expression in response to mannitol, NaCl. And you can see that the cell, this is the serial dilution assay, and these cells which are actually overexpressing this IF from E. coli, from rice, surprisingly, they were able to show performance better in NaCl, in mannitol, as compared to the wild type or the vector transform one. We got very curious that, this is the plant gene. What is it doing in bacteria? Then we extended the analysis to yeast system, and yeast also showed the same thing. There's something very surprising. As you can see, in response to NaCl, in response to mannitol, the cells carrying this particular protein are showing no matter what kind of relation assay you do ODSA, the cells carrying this protein always have a better survival rate. Then we got serious about it. We expressed it in tobacco, and of course, tobacco, the overexpressing tobacco were found to be better as compared to the wild type. And when we expressed it in rice, we were very surprised to see the expression in rice was very, very dramatic, in the sense that if you see the control panel shown on the left side, 2 days, 5 days, 12 days, and 18 days of stress, the one in the middle is wild type. On the left side is the plant where the gene has been knocked down by RNAi, and on the OEIF1, OEIF, this over here are overexpressing them. So you can see under non-stress conditions also, the plant which are overexpressing this gene are showing vigor. This is the health of the plant which you're talking about. And once the plant are healthy, the effect is also seen when you give them stress. You can see that 18 days of stress, wild type is shown over here, knockdown are shown over here, and the one which are overexpressing are shown over here, which is quite logical since it is shown in bacteria, in yeast, in tobacco, it's shown in rice as well. And then at that time, Professor Govindji, who is also known as Mr. Photosynthesis, the world-known leader in photosynthesis, he was visiting our laboratory. He got interested in seeing the phenotype very interestingly. Then he did a lot of experiment with the student. He himself worked with that. And what he told me, using OJIP experiment, using IRGA and PAM, HandyP, several instruments we use. What we finally concluded, to cut the story short for the benefit of student, that the plant which are overexpressing this cytoskeleton protein are having much more healthier, much more sturdy, much more stronger chloroplasts. The membranes are not subject to leaky nature, even in the presence of NaCl. And the cell itself is showing robustness when you overexpress this particular gene. So that's how we could correlate that intermediate filament is very, very important. In a building, what you see, you have iron pillars, then you have the bricks and the cement. But the iron pillars, they determine the strength of the building. And this is exactly what is happening in this case. If you overexpress this iron pillar, you have a stronger building, and which is actually resulting in higher photosynthesis, more sturdy nature, even in presence of NaCl, and more yield over there. When we actually continued the experiment, we analyzed them for the yield. I'm very happy to say that under salinity, the middle one is wild type. On the left side, you have the knockdown plant. On the right side, you have overexpressing line. Under salinity, under high temperature, these plants were again giving almost 35% higher yield as compared. And under drought condition, which the picture we don't have, the knockdown plant did not yield anything. The wild type plant resulted in very small amount of yield, but the transgenic plant were able to recover almost 80% of the yield. That's the beauty of overexpression. That's the beauty of genetic engineering. And this this all came from the allele mining. This gene was be isolated from Pokali, the land race. We are doing allele mining from several other plant species, which is Sweda, which is zero halophyte, from saffron, from pitcher plant. A lot of publications from our lab have actually shown the importance of allele mining. The last part of my story, next three slides, since it is a talk we are talking about nutritionally rich rice also. We have generated lysine-rich rice. Lysine is an amino acid which is very deficient in rice. We always mix up rice with pulses. The importance of pulses have been already been shown just to have khichdi or balanced diet. So there are multiple pathways involved in that. We studied the pathway for the synthesis of lysine, which is the rate limiting step. So we decided to overexpress one gene which is involved in biosynthesis of lysine, and one gene we wanted to knock down, which is involved in degradation of lysine. But the problem is that this is the auto feedback loop. If you have more lysine, there is a feedback inhibition. That means the enzyme, which is DHDPS, dipicolinate synthase, is actually feedback sensitive. So we needed to modify this particular gene. So we looked at the literature, did some modification, and with the help of endosperm specific promoter, several constructs where we hooked them with the pCambia construct, with the 35S promoter, a lot of things we did, at least 20 different constructs we generated. And finally, we were very happy to see that one of the constructs where we overexpress one gene and knock down one other gene is showing almost 50% increase in lysine content. That's something remarkable. Having high lysine rice is a boon. We know that many people in African countries have only access to rice. And if we can make some of these products available to the people, the lysine deficiency is very, very important, because once you have lysine deficiency in your food, it causes a lot of diseases, including kidney failure, and many other diseases which time doesn't permit. And incidentally, since the lysine is an amino acid, it is a rate-limiting amino acid. When you overexpress this lysine, there is overall increase in protein content. I think I have one slide to show you. And since it is overall increase in protein content, the transgenic plant was showing tolerance to salinity as well. And I think and when you look at the fold change of many of the gene of DHDPS, and they show molecular analysis, and this is where we say that the transgenic plant having high lysine and higher amount of protein have better performance under stress condition. This is the wild type, and this is the high lysine content. There is a good correlation between high lysine and high protein leading to high stress tolerance. So there is a beautiful story which we have reported for the first time. All this is published. In case students have more interest, they can log into it. These are some of the details which we have proposed, that how this high lysine is actually affecting and controlling the overall protein synthesis. Because once you remove the bottleneck, which is actually lysine, then you can achieve you want to achieve. So 58% lysine we could increase. We have patented this. And this brings me to thank all the lab members, the previous lab members and the continuing lab members. Some of them have been listed over here. Ratnakaran, Gautam, Sumita, Anita, and so on. And the fund is available from JNU Internal Fund, DBT, DST, International Fund from Sweden, from Vienna. And this is my email address I have listed over here, ashwaniparikh@gmail.com. And I'll be very happy to take questions now, or if somebody would like to send me the question over email, it will be my pleasure to answer. And thank you very much, organizers, Dr. Anil Gupta, for giving me this opportunity, Dr. Chaturvedi. I was very happy. I'm very happy to address to the question. Thank you very much for this opportunity.
Thank you, sir. Thank you, sir, for such an informative talk. Your presentation epitomized everything good in an oral presentation. It was just beyond excellent. Thank you for putting your heart and soul into the lecture. Thanks to all once again, sir.
My pleasure.
In continuation, we have with us one of our distinguished speakers, Dr. Ventriventan Mani. I would request Dr. Anshuman Singh to give a brief introduction of sir.
You ask some question, or Dr. Nishant Bhanu, some question from the audience.
If there is any question from the audience, they may ask.
Can I only make a comment?
Yes, sir.
Hello?
Yes, please. I think I'm
listening. Am I audible?
Yes, sir, you are audible, sir.
Yes.
You are audible.
Thank you, Professor Ashwani Parikh. It's only a comment, and particularly to inspire the youngsters, those who would have listened to his talk. that how excellent was it. And he covered all basic aspects as well as the applied value. He, made excellent contributions from his work. And therefore, he was awarded so heavily by none other than our Honorable President of India. So I think I'm very happy to also listen to your talk, Professor Parikh, and would like to actually, make sure that all those youngsters may kindly give a feedback to both Dr. Chaturvedi and Dr. Anil Kumar, or Dr. Anil Kumar especially, about what they learned from his presentation, actually, because then only it will inspire them more. And let there be a dialogue. Actually, you don't find an example like this again and again, as we have from Professor Ashwini Parikh, who is heading such a large institute, at this young age, with excellent contributions both at national and international level. So kudos to you, salute to you, Dr. Ashwini Parikh.
Thank you very much, sir. Thank you. Thank you very much. You're always encouraging and supporting. Thank you very much.
Thank you, Dr. Parikh. Indeed, I have a small question or query. I would like to ask that Pokali and IR64, are they having a difference in the date of maturity? Because
Yes.
I found that whenever the date of maturity is lower, then they are found to be more resistant, more tolerant, and they are also giving more nutritionally dense, particularly micronutrient like calcium or maybe other important micronutrient. So what is your opinion on this?
So Pokali is actually, I would say, a late maturing rice. While IR64 can mature within 110, 120 days, I'm talking about my own campus, Jawaharlal Nehru University, but Pokali typically goes up to 150 days. So, and the plant is so tall, sometimes if you go to the field condition, from a distance, you can say that this is Pokali, but this goes up to 7 feet as well, which actually is, I think, primary reason the yield is less, because a lot of photosynthesis is actually being diverted to, vegetative tissue. But yes, there are differences in maturity date, and Pokali is a very late maturing rice as compared to IR64. IR64 has been bred for early maturity. All of us know about it. It's the highly cultivated rice being grown in more than 200 countries now. So there's a lot of desirable character, including the date of maturity as well. Thank you very much.
But actually, I found that whenever a date of maturity is less, then they are nutritionally superior. So I don't know about in this case, date of maturity is higher. And but I found that many of the stress resistant genotype, they are basically having more nutrition, and their organoleptic quality is much, much higher. So this is another very, very important area that people have to work it out. What is the relationship of tolerant versus the nutritional superiority?
So I think since I'm in the institution where such facilities are available, I think you have given us the direction to look at the seed quality between IR64 and Pokali. Perhaps that's a new direction we can work on with your advice. Thank you very much.
Thank you, sir. Any more questions? Any more queries? If anyone wants to ask, sir.
I have a small question from Professor Ashwini Sahab. First of all, I would like to thank you for such a talk. You are doing this now, allele mining. So out of many methods of label, which one is the best one for doing allele mining? And also cost effective? Kindly.
Your voice was choppy. I could not get the question properly. If someone else can repeat for me, his voice was choppy for me.
He's asking, you have used many omics approaches for identification of genes or alleles.
Okay.
And which method is found to be more easy and also cost effective?
Oh, okay. For allele mining, these omics approaches are not good. Omics approaches can only give you differentially accumulated metabolite, differentially accumulated protein, differentially accumulated transcript. But for allele mining, you actually need to go for sequencing of those genes, specifically your interest. But nowadays, SNP genotyping can also give us allele mining. So I think the new tools are coming up in a big way. and which are much more efficient. In that case, you can do genome-wide SNP analysis and look at the gene or genome of your interest or particular part of a chromosome for do allele mining. But if you have interest in specific pathways, you can fish out the two alleles from the two respective genotype and then do compare the sequences as well, both at the amino acid level as well as the gene level. So that kind of analysis can easily be done.
Yeah, I mean to say tilling approach.
I'm sorry, I can't.
You are adopting tilling approach. Yeah. You are adopting tilling approach.
Yes, tilling is one approach. Yes, tilling is one approach. Of course, it is a wonderful approach, and a lot of people are using. We have not used tilling. Rather, we have gone for MuteMap Plus, which is much more advanced now. That's the kind of approaches we have used. MuteMap and MuteMap Plus are the approaches which we have used. Yes, tilling is a wonderful approach. I have no hesitation in admitting that.
Okay. Thank you very much, sir. Thank you very much.
Pleasure.
Bye.
Thank you, sir. Any more questions?
Now invite another speaker.
Okay. So I would request Dr. Anshuman Singh to give a brief introduction of Dr. Vetri, sir. Sir, please.
Thank you, Dr. Nishant. Dr. Vetri Venthan, he's a senior scientist working in genetic resources in a gene bank research program, Genetic Gains at ICRISAT, Hyderabad. He obtained his PhD in plant breeding and genetics from Tamil Nadu Agricultural University, Tamil Nadu, India. He is involved in gene bank activities on sorghum and 6 small millets on subjects such as germplasm characterization, regeneration, conservation, and documentation. His research activities focuses on enhancing use of germplasm in crop improvement, such as forming representative germplasm sets of core and mini-core, germplasm gap identification, evaluation, and identification of trait-specific germplasm sources, developing genetic and genomic resources, and so on. Having expertise in plant breeding and genetics and plant genetic resources, he has over 50 publications, including 20 research articles, 1 review, and 5 chapters in books. And he has also edited 1 book and 4 training manuals. So I would like to invite Dr. Vetri Venthan to deliver our talk. Over to you, sir.
Thank you, Arsman. Yeah, I would like to share my screen. Is it full screen now? Can anyone confirm me? Am I audible?
Yes, your screen is there, but you have to make it in slide mode.
Okay. Is it okay, sir?
Yes. Okay. It is okay.
Yeah, okay. Thank you very much, sir. So, first of all, I would like to take this opportunity to thank Dr. Anil Kumar, Director Education, Dr. S.K. Saturvedi, Dean, and Dr. Arvind Kumar, and other members of the organizing committee for organizing this wonderful event, and also for providing this opportunity. And also, I would like to thank my previous speakers, Dr. K.C. Bansal and Professor Ashwani for their inspiring speech. So I would like to take you back to plant genetic resources. So myself, Vetri Venthan, already briefed, Anshuman already briefed about me. So I'm working on sorghum, palm millet, and 6 small millet germplasm resources on conservation and also enhancing the use of germplasm in crop improvement. Today, I will be talking about plant genetic resources characterization and then allele mining. So the PGRs is a key to drive any crop improvement program. So all of us know already Dr. Bansal and also many other Chaturvedi sir also mentioned many things about plant genetic resources and their importance. A large variability exists in the germplasm for various agronomic traits, biotic and abiotic stress tolerance traits, and also for the quality traits. The very good phenotypic and genomic investigation and identification of the naturally occurring allelic variation at candidate genes controlling key agronomic traits which has potential application in crop improvement program is very important for breeding climate resilient and nutrient dense crops. So I will start with plant genetic resources for the interest of the students. So any material of the plant origin that includes reproductive or vegetative propagating materials, that contains functional unit of heredity that is called as plant genetic resources. It can be seed, or it can be plants, or it can be the tissues, or DNA. All these are considered as plant genetic resources. And these plant genetic resources contribute significantly to these sustainable of the United Nations, particularly to end hunger and also to end poverty and other sustainable development goals also. And these plant genetic resources are conserved in two ways, majorly two ways only. So first one is in situ conservation, that is conservation of the plant genetic resources within their natural habitat, that is called in situ conservation. That includes natural parks, biospheres reserves, and also on-farm conservation. So farmers considering land races over hundreds and thousands of years, that is also a kind of in situ conservation. And the next one is important one is the ex situ conservation of these plant genetic resources outside their natural habitats under controlled environment condition, that is called as ex situ conservation. So this is just for the interest of the students, just I am briefing. So this ex situ conservations includes the gene bank, DNA bank, pollen bank, also tissue culture. So all this conservation of these materials under the controlled condition is known as the ex situ conservations. These slides already explained by Dr. K.C. Bansal sir. So globally, there are about 7.5 million germplasm accessions are conserved in more than 1,750 gene banks, and there may be about 25 to 30 unique germplasm across gene banks, and about 10% may be the wild species among these, and 11 CGR gene banks, that is conserving the largest portion of the genetic resources of the important crops, and it is the main source of the germplasm for the research globally and contributing to the food security. And if you take example in millets, globally there are about 220,000 germplasm accessions are there in millets alone, and in pearl millet there are about 73,000 germplasm accessions that are conserved in 57 gene banks, and in finger millet also we have a very good number of accessions. Finger and foxtail millet also we have a very good number of accessions, about more than 40,000 germplasm accessions are there. And in other millets also we have a very good number of germplasm, but the minor millets we have less number of germplasm, maybe less than 10,000 or even less than 1,000 also available. That needs to be collected and conserved before we lose them forever. And at ICRISAT Gene Bank, so we are working on at ICRISAT Gene Bank, we are conserving 11 crops germplasm accessions. Currently we have 129,000 germplasm accessions that are originating from 144 countries. And as of now, we have distributed about 1.5 million seed samples to researchers globally and distributing the diversity in the service of the global farming. And coming to the allele mining, which is very important, almost already many told about allele mining, what is the importance. There are some of the important approaches through which we can do allele mining in the germplasm collections, that is genome-wide association mapping and genomic prediction. And the third approach is tilling and eco-tilling approach. And for these approaches, the important key is phenotypic characterization for the success of the genome allele mining. So we have large number of germplasm in major gene banks like national and international gene banks. Is it possible to characterize the germplasm for important traits? Is it possible to characterize the total collection? So we need to just think, if yes, for which traits we can characterize. Already Dr. Bansal sir said about 20,000 germplasm accessions that were characterized at one site. That is very great, so that the researcher can see the diversity on form and they can select the very different traits. If you have very good field and then if you have facilities, maybe you can. If you have very good facilities like NIRS and XRF facility, then we can do very good high throughput screening of the nutrition traits and also fodder quality traits you can do. And also nowadays is sick plot and glass of screening of the diseases and herbicide tolerance also, for example, that can be screened in large scale, and mechanical harvesting, for example, in chickpea and any other crops that can be screened in large scale, lodging and water logging. These are all some traits that we can explore and we can do some large scale germplasm evaluation if facility is there. But all these traits needs a lot of investment and a lot of manpower and resources. If you take example in sorghum, actually, out of we have at Gene Bank we have about 42,000 germplasm accession in sorghum conserved in the Gene Bank, and these 42,000 germplasm accessions are characterized for morphoagronomy traits following the descriptors, and these descriptor traits are available in the database in the ICRISAT website, and you can go to this database and then you can see different traits, and then you can choose. You can also filter the accession for different traits like early maturity you can choose, or tall plants you can choose, or different country ways you can choose, so that you can make a subset. That subset can be utilized in the allele mining program or for the phenotypic and genomic characterization. And there is one more global germplasm database is there, that is called Jhansi. There also we have lakhs of germplasm accessions are there. You can see here so many germplasm accessions are conserved from the national and international gene banks that is available. Passport and characterization is available. And there are many characterization and evaluation subsets also available. So the researcher can go to this and then they can explore the germplasm, and then they can also request germplasm for their research. And there are many subsets also developed that is uploaded in this Jhansi, so that is also available. So the researchers can go to this website and then check, for example, whatever germplasm subsets we have developed at ICRISAT, it is available in this Jhansi. So this is one of the important resources for the researchers who are working in the germplasm. And if it is not possible to characterize, or very difficult to characterize, or if the facility is not available, and if you want a small number of germplasm, then the options are like development of the diversity representative subset, like core or mini core collection. You can develop a diversity representative subset following different approaches, or there is another approach is there for the identification of trait specific sources. That is called focused identification of germplasm strategy. And it is the concept of the core collection is very old, and it was proposed by Frankel in 1984. The core collection represents 10% of the total collection conserved in the gene bank, that represent the diversity of the maximum diversity of the collections conserved in the gene bank. So for developing core collection, you can use passport data or characterization data or taxonomical data. You can use, in addition to this passport and taxonomical data, you can also use the phenotypic characterization or evaluation data. And also if your genotypic data is there, you can use this genotypic data also to develop a subset that represent the entire maximum diversity of the total collection. And then that subset can be utilized for the germplasm characterization and evaluation at multiple locations and multiple years, and also for the genomic investigation to map the traits and using the crop improvement program. So sometimes the core collection also, sometimes it's large in size, therefore we may need to reduce the size of the core collection. So we can develop, so the concept of core collection was proposed, concept of mini core collection was proposed, that was 1% of the entire collection and 10% of the core collection. So there is a steps to steps to be followed for the development of subset like core collections. First, group the accessions, group the entire germplasm into some different set of groups, like using the taxonomical, morphological, or geographical, or sometimes the mostly the racial characteristics or regional different regions can be grouped separately. For example, if 40,000 germplasm is there, and you can group the germplasm into different races. If you take example in sorghum, there are 5 races are there, and 10 hybrid races are there. Maybe you can consider that as a group and then make a grouping, and then using the taxonomical and also characterization data, molecular data if available, then we can do diversity analysis and develop a cluster, and then from each clusters we can develop 10% of the accession to develop a core collection. So for developing mini core collection also, we need to follow the similar steps to develop mini core collection also. Once while selecting the germplasm from the clusters, you can follow some random sampling approach or the preferred sampling approach also. Once we develop the core collection, then we need to ensure that the core collection represents the entire collection in terms of the mean of the different traits, and then variance of the different traits, and then range of the traits and diversity. All these things we need to ensure that the core collection representing the entire collection, maximum diversity it should ensure. There are many tools and softwares are available for developing core collection and mini core collection, like window-based as well as R-based softwares are there that can be developing this core and mini core collections. Following this approach, we have developed a core collection in all our 11 crops that are conserved in the gene bank. If you see the core collections, number of accessions in the core collection ranges from 75 to more than 2,247 accessions are there in the sorghum, and sometimes number of accessions in core collection is largely for most of the crops, like if you see here. Therefore, we have developed a mini core collection so that mini core collection can be manageable in size, that can be extensively characterized and evaluated for the morphoagronomic quality and nutrient, and also biotic and diabetic stress tolerance. So you can see here the maximum number of accession in the mini core is 242 accession. This is manageable in size that can be utilized in the germplasm evaluation activity. So using this core and mini core collection approaches, we have identified a lot of germplasm accession that are very good for different traits, like and also many germplasm accession that are sources for multiple traits also. You can see here germplasm accession and their traits that are contributing. And here, if you take example of this IS 4698, and this is resistant to spotted stem borer, shoot fly and aphids, and also it is giving good yield also. This type of germplasm can be directly utilized in the crop improvement program and also utilized in the genomic investigation to map the traits and the genes responsible for these traits. So similarly in chickpea also we have done a lot of germplasm characterization activity using the mini core collection, and then trait specific sources have been identified for biotic and diabetic stress tolerance and agronomic traits also, and also for the both agronomic and nutrient traits also. And once you develop the core collection and we have the trait specific sources that are identified from the core or mini core collection, if you need additional number of accession, then you can go back to the collection, core collection, and the cluster from which the mini core collection was selected. If you need additional source of accession, then we can also go back to the total collection or entire collection from where these accessions were selected. So therefore, it will increase the probability and chance of identifying new source of accession. More probability of identifying the additional accession will be higher. So another approach is called the focused germplasm identification strategy, and it is well known that the survival of fitness is the concept. Here, we all of us know that environment strongly influences natural selection. So therefore, it increases the chance of finding the adaptive traits in that particular environment. For example, if any environment that having high drought occurrence, frequent drought occurrence, and also low rainfall area, the land races that are surviving under that situation can be having adaptability to that trait. For example, the Pokali that is salinity tolerance that is adaptable to that salinity tolerance. If any germplasm from the coastal area that is surviving and also producing good yield, and also that can be utilized, that can be used, that information can be utilized to develop a subset. That is the approach here, focused identification of germplasm subset. So here, this approach matches plant traits with geographic and agroclimatic information of the places where these germplasm accessions are grown. So utilizing this information, actually there are two approaches are there. So filtering approach is there and the modeling approach is there. For filtering approach, we need to have deep understanding about the optimal condition of the expression of the traits. And if you have the deep understanding and the information under which condition the particular stress happens, then we can use that information, and then using the coordinate information, we can pick the germplasm that are from that particular environment. That is called a filtering approach. If you have some, in case of modeling approach, if you are already having some characterization and evaluation data, you can use that data and then make a prediction, and then you can select a subset from this modeling approach. I will just tell one example of this sun pest in wheat. There are more than 16,000 germplasm accessions are there. To develop a subset of fixed subset of 534 accession, so there are filtering approach was followed, and here you can see out of 16,000 accessions, so the materials from the coordinates from 30 to 45 latitude and longitude were selected initially because occurrence of the sun pest is more in case of this latitude and longitude. Therefore, it was selected. Then next step, they excluded accession from China, Pakistan, India, because there is no report of this sun pest occurrence there, or it is recent report was there. Therefore, there may not be availability of that adaptation traits in that the germplasm. Therefore, they excluded. So they came down to 6,328 accessions. Again, they excluded accession from the dry environment, that is rainfall below 280 mm per year, because the dry environment does not favor the sun pest occurrence. So they excluded accession from this dry environment. Again, they also excluded accession from the winter temperature fall below 10 degrees Celsius, because this environment also does not favor occurrence of this sun pest. Therefore, after making all this filtering, and then from out of 16,000 accession, the researchers got 534 accessions. So this 534 accession was evaluated for the sun pest, and then they got very good resistant sources, 9 resistant sources they got. Instead of screening the entire germplasm, they filtered to the subset. They developed a subset, and that subset gives higher probability of getting more number of this trait. There are many studies are available on this focused germplasm identification strategy on different traits. There is, I just want to make a small comparison about the core collection and focused germplasm identification strategy. This core and mini core collections represent the maximum diversity of the total collection that is conserved in the particular gene bank. But the focused subset, fixed subset, does not represent the entire diversity, because the subset is developed for the specific traits, not for the diversity. And the core collection, in the core collection, we can identify multiple traits also. You can identify biotic, abiotic stress tolerance, or for the quality, or many more traits. That is possible in case of core collection. But in case of focused germplasm subset, the subset was developed only for the specific traits. Therefore, the chance of getting other traits may be there, but it will be less only. And here, in case of core and mini core collection, there is a lower probability of finding desirable traits, because this core collection represents the entire collection in terms of diversity, and we may not find the desirable traits. But here, in case of this focused germplasm identification strategy, high probability of getting the desirable traits, because this subset was developed particularly for this trait only. Therefore, you will be having higher probability of identifying the desirable trait of interest for which the focused germplasm subset was developed. So these kind of subsets can be very good genetic resources, germplasm resources for the allele mining and genome allele mining of so mining the allelic variation and identification of the trait-specific germplasm and candidate genes. So here I mentioned there are 3 approaches that mostly we can utilize in the germplasm characterization. So for these approaches that can be utilized for the allele mining in the germplasm, one is genome-wide association mapping, and then genomic prediction, and tilling and eco-tilling approaches. So this is, I don't want to explain here. This is very well known concept, the genome-wide association mapping. Here we need the association mapping panel. The subsets that we have developed in the core, as a core or mini core subset, that can be a wonderful starting material for this study. And that material needs to be phenotyped and genotyped using this phenotypic and genotypic information, and then population structure and family relatedness. So genome-wide association mapping is done, and then this marker trait associations are identified. In Proso millet recently, we sequenced 200 germplasm accession using genotyping by sequencing approach, and then we also phenotyped for the agronomic traits and grain nutrient traits, and then we identified marker trait association for this productivity and also grain nutrient traits. Recently, we sequenced 310 accessions of finger millet, that includes the core and mini core collection accessions, and also some of the released varieties and elite breeding lines. So we formed a diversity representative subset of 310 accessions, and this diversity representative subset was sequenced through dot sequencing approach, and then we characterized we analyzed the gem blossom for the population structure and diversity, and then we also the same set of gem blossom we assessed for the agronomic traits and grain nutrient traits. You can see large variabilities there for the different grain nutrient traits, particularly for finger millet is particularly very much important for the calcium, and you can see very good large variabilities there for calcium and also for the other traits also. And so this and the same set of material we also characterized them for quality traits, fodder quality traits also, because finger millet is one of the important crop for the fodder also. Therefore, we assessed the fodder quality of these successions. And then, so using these materials, we are going to do the genome-wide association mapping and genomic prediction for the identification of the genes and marker trait association. So using this sorghum mini-core accession as an association mapping panel, we have done a lot of genome-wide association mapping, and then we mapped different traits like we identified marker trait association for the different diseases like for the different. So we have identified.
Please mute yourself.
चैट खोलिएगा थोड़ा चैट खोलिए प्रीति का गंदा बात तो नहीं किए होंगे आप.
Please mute yourself.
Thank you very much. Okay, so coming to the another important approach after genome-wide association mapping and then genomic prediction is largely used in gene bank collections. Here the core collections, like as I mentioned, the core collection can be the best population for the genomic selection, and it can be this genomic selection can be utilized as a some lines or edits are coming. Okay, so for this genomic prediction, we need a training population. The training population can be the subset that we have developed. So this training population need to be phenotyped as well as genotyped for the traits of interest, and then we need to do we need to train the GS model, and then after the prediction, then we can take another set of gem blossom which that only having the genotyping information and to predict the breeding value of the traits. So there are many studies available on the genomic prediction of gene bank collection on wheat and many other traits, many other crops like wheat and chickpea also recently in the ECRISAT we have done. So in wheat for the rust, grain nutrient content and heat and drought tolerance also there, and for the chickpea, for the productivity traits we have done at ECRISAT led by Dr. Raju Vashni. So this study already very well explained by Dr. K.C. Bansal sir. Actually, at ECRISAT, we have Dr. Raju Vashni and the team sequenced more than 3,300 accessions of chickpea. That includes more than 3,171 accessions of cultivated, and also wild accessions. So 195 wild accessions are there. So here they performed the genome diversity analysis, and also they made, and in addition to this basic work, they also made a genomic prediction for the yield and the yield contributing traits. They found about genomic prediction value of about 0.9 accuracy, genomic prediction accuracy of 0.9 for the plot yield, and also for the underseed weight also there was a good prediction accuracy was found. And apart from this genomic prediction, they have identified superior haplotypes for productivity traits such as underseed weight, days to maturity, plant height, pots per plants, and also this plot yield. These superior haplotypes can be utilized in the breeding program for the haplotype-based breeding crop improvement. So another study in soybean from the USDA Gene Bank. So they have about 90,000 gem blossom accessions. They took about 465 accession, diverse accession, and then they phenotyped that diverse accession for the white mold, and also they also genotyped using the sequencing approach. And then out of this 465 accession, they took 352 accession as a training set, and then 88 accession as a validation set, and also they performed the GS training model, and they performed the genomic selection. and the genomic prediction accuracy was about 0.4 to 0.43 to 0.47 in the field data. And now, once we have this information, then we can apply this genomic prediction in the entire germ plasm collection when we have only genome sequence information is available without doing any phenotypic characterization. Once we identify the good accession that are having very good promising traits that can be validated. Another study from the Uganda Gene Bank is on bread wheat on seed traits. They also have done genomic prediction, and they found the prediction accuracies of 0.64 to 0.74 for the seed width. So these are all some of the examples of genomic prediction, particularly in the gene bank collection. In another studies also there in the sorghum, they used the genomic prediction for the biomass traits. They genotyped 296 genotype 296 accession, and also they performed a genomic selection approach using the 580 as a validation set, and they found a prediction accuracy of about 0.292 to 0.56. So these are some example. And then another approach is eco-tilling and genome sequence, gene sequence based approach. Here, the stilling and eco-tilling approaches are used to identify allelic variation within genes of interest to isolate the desirable traits. In case of eco-tilling, the natural mutants are used. Natural mutants in case of the germ plasm diversity and natural population can be used here, and then here the mutagens are used to create variability, and then gene-specific markers are used to do PCR and then identify the allelic variation within the genes of interest to isolate these desirable mutants. There are many studies are available on these approaches on different traits and in rice, wheat, barley, sugar beet, and also potato. A lot of studies are available. So these are the some. I just wanted to brief about this, what we have done and what other gene bank researchers have done in the genomic prediction and allele mining of the gene bank collection. So in brief, germ plasm is a key to drive any crop improvement programs. That is very well known that it is the key for the crop improvement. A large variability exists in the germ plasm collections. So what we need is we have very advanced technologies are there, advanced high throughput phenotyping and genomic technologies are there. We need to use appropriate subset and also strategies for the mining novel variations and their implication in the crop improvement. So if researcher interested to get germ plasm from the ICRISAT and also from any other gene banks, they can check their website from ICRISAT. If you want, you can also write to seed request. Email ID is there here. I just put here email ID, and you can send email to seed request and also Dr. Kuldeep Singh, who is head of the gene bank right now. And you can write to these emails and you can get this germ plasm. And you can also go to the ICRISAT gene bank database. There you can see a lot of information available on the phenotypic characterization data and subset. That data can be directly also you can request. So I would like to take this opportunity again to thank everyone for the opportunity.
Thank you very much, sir, for such a nice talk. You are really doing a great job while working on these orphan crops such as millets. Although these crops have some unique features, unique traits, but they are lagging behind when we talk about the mainstreaming of these crops. So yeah, you are doing a commendable job while working on these underutilized crops. And I believe that there must be more tools should be developed for these crops in terms of genomics and those multi-omics tools, so that we can mainstream these crops in future. Thank you very much, sir. So before we go forward, we can take a few questions from the audience. If somebody have questions, please ask.
Good evening, sir. I am Dr. Nitish, working on rice at ICR Canning Town. Actually, I have a question to Dr. Vetrivandan, sir. Sir, in your slide, you have shown us about focused identification of germ plasm strategy. In that, I want to know that if you have like same kind of geographical things located like in different places of the world, and if you have like different germ plasm from there, So from that, how you are going to make a kind of means like core set of germ plant just to use that for like genomic selection or for Jiva study or something like that. Means what portion we can go, or how we can go for selecting that, because at all the places we may have different number of like genotypes or land races from those places or from those areas.
Yeah, okay. Yeah, I got your question. So actually this focused identification of germ plasm strategy, it is involving so if you have any information about the adaptation of the germ plasm from different places, for example, the drought tolerance may be in the location where the drought occurs more frequently, there the germ plasm that are adaptable to that location will be having the drought tolerance ability. So similarly, in many locations and many places in the globe, there will be having the adaptations. Based on the available, the latitude and longitude information of the gene bank land races, we will be using that information to retrieve that soil and climate and environment information from the websites. That information will be used to extract the germ plasm subset. That subset will be, instead of evaluating the entire germ plasm, we can evaluate the subset. So here we are going to get higher probability of getting promising germ plasm. So there are two approaches are there. One is the filtering approach. If you have some information about the trait. If you don't have information about the trait and then you know some germ plasm already evaluated for the trait, and from this germ plasm coming from some particular region, that kind of similar environment we can also look for and then make a subset. This is what modeling approach. The first one is filtering approach. This both approaches you can make to make a selection.
I think Dr. Mani, thank you very much for your presentation. I would like to ask, like when you are making a core and mini core of different accession, it is not right way that we have to go for genomic selection where we have to see the test and reference panel. So I think that will be more appropriate comparison to your core and mini core. What is your opinion on this?
So the concept of core collection is because to enhance the use of germ plasm diversity in the crop improvement. So once you have that subset core or mini core collection, you can use that subset and you can do genomic characterization and phenotypic characterization, and then you can make a lot of additional sources from the entire germ plasm. It is a gateway to search the germ plasm from the entire germ plasm. You can use molecular data and phenotyping data to make a reference set.
I think both back and forth approaches are required.
Yes,
so that you can go more precise characterization of these useful germ plots.
Yes, yes, yes.
Okay. Thank you.
Okay, so in addition to this, sir, actually, like most of these millets, they don't have like fully annotated genome sequencing. Right, sir?
Yes, yes, yes.
So like how you deal with, like if you go behind some specific pathway specific to millets, like, but we don't have such information if we want to, like sequence those genes, or if we want to clone these sequence in some other, like C3 or C4 plant. So what are the like problems you are facing while dealing with these issues?
Yeah, actually these crops are underutilized crop in terms of the area as well as their utilization, and also genomic resources available in these crops are also very poor, very low.
Right. Sir.
Recently only the finger millet genome sequence was genome was sequenced, and the foxtail millet is very well characterized crop. We have very good genome, reference genome is there, and for barnyard millet also one species reference genome is there. And other crops we need to make the sequencing we need to do. If you do not have sequence information, reference genomes are there, then if reference genomes are not there, then you can also genotype using genotyping by sequencing approach, because that does not need any reference genome. So you are going to assess the diversity, and also you can look for the homology with the other genomes. For example, if you have rice, you can also compare with rice, and for foxtail millet we can compare with rice and other crops, and for proso millet we are comparing with the panicum virgatum, that is switchgrass, which is related one. So through that way, we can go and compare it. And also recently I did dot sequencing. That also does not need any sequence information. So that will give very good platform for genotyping these underutilized crops. Once you have the reference genome, then again you can also do mapping and then do further work.
Right.
Work and other works we have not done, so I don't want to come on.
Right. Okay, so in this series, I take this opportunity to introduce Dr. Swarup Parida among the audience. So Dr. Parida is currently a staff scientist at National Institute of Plant Genome Research, NIPGR, New Delhi. He obtained his PhD in biotechnology from Jamia Hamdard University, New Delhi. He gained his postdoctoral research experience at National Research Center on Plant Biotechnology, New Delhi. His research area is on plant molecular genetics and genomics, and he is mainly working on genomics of seed and pod traits in chickpea. He has published more than 100 articles in national and international journals. He conferred several awards such as Samantha Chandrasekhar Award in 2018, Odisha Young Scientist Award in 2014. He is also the recipient of Nasi Reliance Industries Platinum Jubilee Award in Biological Sciences. He is also the fellow of National Academy of Agricultural Sciences and National Academy of Sciences, India. So now I request Dr. Parida to deliver his talk. Over to you, sir.
Am I audible? My slides are visible?
Yes, Dr. Parida. Good evening.
Yes.
Thank you.
Yes, sir. So good afternoon, everybody. So first of all, I like to thank Professor Anil Kumar for giving me this opportunity, and also respected Dr. Chaturvedi, Professor K.C. Bansal, Professor Ashwini Parikh, and Dr. Vetri Veran. And thank you all, and on all the participants, and good afternoon to all of you. So today, this webinar, this title when I seen, so this is that the new dimension in crop improvement. So that is from Allele mining to genome editing. So in context of this webinar concept, that is the new dimension in crop improvement. So in this direction, I want to put this title, that is next generation molecular breeding. So because this molecular breeding we are using, traditionally we are using classical molecular breeding, classical genetics, and also different kind of molecular breeding. So how now, due to this technological advancement, how this next generation sequencing or several kind of new approaches, which how it will be useful, this molecular genetics and also breeding to give new allelic variant. So that is the concept of today's discussion point of today's webinar. So how new allele, not only the new allele, how the superior allele. So this superior allele, which is giving you the trait which we are looking for, which is modifying, and also it has less pleiotropic or epistatic effect to other trait, and it is giving also higher yield. So in terms of that, it can be called as a superior alleles, and how to mine that allele to utilize in crop improvement in terms of marker assisted selection or genome editing. So I will highlight some of our research work on with regard to this. So coming to this slide, so I need not to explain this. So as we know that we continuously want that this production and all this climate change, population growth. So that's why we try to develop the climate resilient, abiotic stress tolerant, and different kind of nutritional improved. So that was already discussed in this webinar by previous speaker. So I not go into detail but I want to highlight one of these points. So as you can see here, before Green Revolution, as you can see, it's in 10,000 years we produce 1 billion ton before 1960. But after the Green Revolution, you can see in 40 years we double the production and yield. So therefore, this technological advancement, but technology adoption of suitable technology, how it has impact to our food security. So that I need not to explain, because in Green Revolution, you know that is wheat and rice, that several kind of disease tolerant and high yielding variety was developed. so that we get higher yield, and not only in terms of classical genetics to develop hybrid variety, but also adaptation of different kind of new agronomic practices. So that also fertilizer application, NPK application, so that all this help us to develop high yielding variety. So that I will not discuss much, but so here this major point is how the classical genetics has developed high yielding variety, and adaptation of new technology, how it has helped us to give the higher yield and productivity. So that's the importance of technology has come. So why we need to adopt the new technology. So coming to this slide, so how this after green revolution, the technological advancement has occurred. So that we need to understand first. So as you can see here, in 1960, the green revolution has occurred. After that, as you can see here, the DNA sequencer, this first generation sequencer that Frederick Schenker 1975 it was discovered. Then, as you know, that PCR, then DNA fingerprinting, then random molecular markers, then several kind of sequence-based molecular marker in this 1992, like SSRs, SNP. So that I will not discuss. Then the first gene was cloned for rust resistance in barley in 1999. So as you can see here, I have already told this first generation sequencing. So utilizing the map-based cloning approach and the whole genome shotgun approach, utilizing the first generation sequencing approach, as we know that from 2000 to 2005, as you can see, the first plant genome, Arabidopsis genome, then rice genome, then also human genome, all has been decoded in the first generation era. But after 2005 to 2010, as you can see, this is we call as a next generation sequencing era. We got several kind of platforms. So like we know that Illumina HiSeq, ABI Solid, Iron Torrent, and several kind of short read sequencing platforms was developed. So then from 2011 to 2020 till now, we are going up to third generation sequencing like BioNano, PEG, BioNano4. But what is the difference between these three era? So this first generation sequencing era, you can go to getting long read, low data output, mostly in case of MB data. But in next generation, we are getting short read, high data output. Mostly it's a terabyte of data, but in mostly GB data, because short read means mostly 100 to 120 base pair sequence and also millions of sequence read. But complementing to this technology, now you are in the third generation where we are getting long reads also. So it is more than 10 KB size fragment and 10 to 20 KB, and also high data put, millions of reads we are getting. So that's why it is now going up to terabyte or picobyte. So that is the technological advancement has occurred. So therefore, utilizing this technology, several crop genome was decoded. As we know that so like cereals genome. So am I audible?
Yes, sir. Yes, sir. You are audible.
So utilizing the first generation, next generation, and third generation approach. So as we have listed, several cereal, legume, vegetable crop genome was decoded, and this list was continuously updating because every day new genome was getting decoded. So, but coming to Indian contribution, so here you can understand what is the significance of adoption of new technology. As you can see here, in case of rice genome, only chromosome number 11, that is long arm chromosome. So that is India was involved for sequencing. As you can see here, several countries was involved in several chromosome, and it has taken 5 years to decode this rice genome because it is a map-based cloning approach and it is a high quality gold standard sequence. So therefore it was 20x coverage. It was sequenced by several people, but it has taken long time and several countries was involved. Likewise, also you can see tomato and wheat. So India is the part of some of the part of the chromosomes, and several countries was involved. So because of this first generation sequencing, so lot of country and long time was required to decode the genome. But as you can see, as next generation sequencing and third generation sequencing has come, as you can see this case of chickpea. So only one institute, 2-3 scientists is able to decode the desi, and also white chickpea in 2 to 3 months, 2 to 3 years time. And also ICRISAT, Professor Rajiv Harsni also involved and decoded Kabuli genome. So utilizing the NGS approach, they can able to decode the genome in the 1 or 2 year time, involving not only one country, only one institute. So that is the power of technology. So as you can see, several genomes was decoded only by Indian scientists. So like pigeon pea, finger millet, pearl millet, tulsi. So like that, as you can see, I have already told that chickpea, desi, and Kabuli. So once you decode the genome, you get their sequence information, so genes information, how many transcription factors, and their in terms of structural genomics information, functional genomics information, in terms of comparative genomics information. All this we can get, but as I have already told, I will not try to explain this more because of this first generation, we are getting less amount of data. So therefore it is narrow, but in next generation, you are getting large amount of data. But the information we are getting same. So we are getting the genomic sequence, transcript sequence, or genes characterized, all we are getting, but only the concept is different. Because in first generation, we are using mostly that clone by clone, that is microsatellite enriched library, cDNA library, genomic library, but now we are doing genome resequencing, transcriptome sequencing, RNA, small RNA sequencing, bisulfite sequencing, and we are getting the same amount of data and quickly. So this was already explained by Professor Bansal, that what is the relevance of pan-genome, but I will explain in shortly. As you can see in case of rice, because of third generation, you can now able to assemble different SS1. So different SS1 has different characteristics for particular trait. So like you can see in SOB1, you can see this FR13A, which is the original donor of SOB1. So where this allele is present, but in Nippon bear, which is the reference genome, we are using continuously for this reference genome, that Japonica Nippon bear. So there this SOB1A, this functional allele is absent. So therefore, we need to sequence more SS1 to create the pan genome. So
लोग हैं भाई, उसको सर्टिफिकेट देना ही पड़ेगा, और नहीं तो क्या, उनके पास लोग ही नहीं है भाई, वो सब.
So here you can see that this reference genome, so some are dispensable genome, it was distributed in several SS1, and some are core genome, so that is present in all SS1. So therefore, we need to utilizing the third generation sequencing. Now you are using PacBio, BioNano, you can generate the assembled data for each SS1. So different kind of SS1s, like in rice, there is Japonica specific, OS specific, and aromatic specific, basmati specific. So that kind of information one can generate. And you can see new genes also. That was already explained by Professor Bansal. So how many genes we got in case of chickpea when we constitute pan genome. But coming to today's talk, so how this next generation sequencing we can utilize in the crop improvement. So that simple slide I have put here. So this is two variety. When you sequence them, so you get several kind of sequences. So if you sequence them and compare them, you get simple sequence repeat. You can see here GA expanded in some variety. It was contracted in another variety. So based on this variation, you can get sequence variations, and also there is SNP variations, and also there is insertion deletion also. So here you can see G by A, C by G, their substitution. So this is called mining of allele. So that is the today's discussion, that is allele mining. So once you mine these alleles, we have to try to associate with a particular trait. So that's why this phenotyping is required. For that's why I put this is the resistant and this is susceptible. So once you mine the alleles, so now you have to genotype them. So for genotyping, there is several kind of tools, that is sequencing tool, sequencing based genotyping tools, like now you are doing GPS, genome resequencing, like 3000 rice genome, and like chickpea 429, now 3326 genome SS1s, and also array based genotyping, like 50K array, 90K array. So that I will give in the next slide some example. So once you genotype in several SS1s, you can get the similar information, sequence variation data. And then the sequence variation data you can correlate with the phenotyping data. So you can correlate like G, C, this combination is present in the resistant variety, but AG is present in the sensitive line. So therefore, the GC can be a new allelic variant or can be a new haplotype to present in the resistance. So if you transfer this allelic various data to allelic data to GC to sensitive variety, if it is low yielding, so it can be become the sensitive may be high yielding and it is sensitive. So you can make them resistant and high yielding. So likewise, one can analyze the data. So you can see if this sequence, it is a particular gene. You can tell that this sequence variation data, but this allelic variation data, G by A data, is present in a particular gene. And this is the allelic variation. And this is the haplotype because the GC, it is the superior haplotype because it is present in the resistant variety. And once it is transferred to the sensitive variety, it is giving higher yield, not pleiotropic and epistatic effect to other trait. So that is the significance of identifying the superior allele. So once you know that this is the superior allele, it can be utilized as a marker for marker assisted selection, or you can utilize this allelic variation. You can do genome editing also. This A can be replaced by G through genome editing or gene editing, or G can be replaced with C. So that kind of analysis one can do once you mine the superior allele which is associated with particular trait. For that, we basically using this kind of data. So if these two varieties, so these two varieties we have discussed today, so that several kind of germplasm accessions are there in the gene bank. So this, if all these lines are germplasm accessions, and this is the sequencing data, so we can calculate their historical recombination. So based on genome-wide association mapping or gene-based association mapping that Dr. Bhattacharya has already explained, and based on that, we can told that this genomic loci is associated with particular trait. So another method is that we know that QTL mapping. If these two variety, it is a parental type, parental accessions for particular mapping population, we can cross them and you generate several segregating line. And similar way, you genotype them, you get the sequence information data, correlate with the phenotyping data, do genetic recombination, and try to identify the QTL. Then once the QTL, then we try to narrow down that QTL to particular gene through map-based cloning or positional cloning through fine mapping. So likewise, once you identify this allelic variation data, your haplotype is associated with particular trait through GWAS or QTL mapping, you can utilize now this allelic variation or superior haplotype for trait improvement, either through genome editing or through marker assisted selection. So this is the basic idea how from sequencing one can really quickly reach to the particular gene and allelic variant. So as we know that we need several kind of accessions, so that I will not discuss today because it was discussed in previous speaker so much how different kind of accessions we select, so that I need not to explain because land races, wild species, and how core and mini core needs to be constituted. So like it was already explained, what is the importance of core and mini core collection. As you can see here, we need accession also, phenotyping data also, and genotyping data. So for accessions, mostly people are using core or mini core, or they constitute trait specific association panel. So this panel by the germplasm one has to phenotype and see their diversity. So that also explained by several speakers. And also once you have this variation, we can phenotype them also in control condition also, and in greenhouse or in field condition or in phytotron, because based on your trait of interest, we have to do that. Not only I can tell that only field is important or phytotron is important, because sometimes both is required to get robust phenotyping data. So that one has to decide based on the trait which they want to dissect. So now several kind of high throughput phenotyping platform. This is the view of ECRISAT, where you can phenotype by non-destructive method, by root scanning. So thousands of germplasm accessions or mapping population derived lines, you can phenotype them. And also there is several kind of, now people are coming model-based approach like thermotolerance, root architecture. So that can be also done through this kind of phenotyping, so that basically we are looking for large number of accession can be phenotyped more precisely in the less time, so that this kind of technique was coming, so that we can get best loci to be associated with a particular trait. As I have already told, you can generate mapping population also like that. So as germplasm accessions, so like that you can cross them, and in reels you can see their trait segregations, and each genotype you can isolate DNA and genotype, as I have already explained. And also we are now looking for mutagenes population also. So chemical mutagen or radiation-based mutagens. So as you can see here, this is the rice variety Nagina 22. When it is EMS mutant, it is become dwarf. So you can do like already Dr. Petri has told, it is stealing approach. You can approach stealing approach, or now this is the quick NGS based approach that is new-to-map approach, by which you can identify the genes or loci or allelic variant which is associated with the particular trait. Like here, can be plant height. So likewise, also there is several platforms are now available to advance the generation for like genome edited line, transgenic line, also breeding line, so that you can generate F7 or F6 generation in 2 or 1 or 2 year time, so that you can do implement discrete breeding technique by which you can shorten the generation time and accelerate the breeding and research program. So coming to the different kind of genotyping tools in SSR, mostly we are using the gel-based assay. So it is visual estimation. Here you can see one SSR. If you genotype in multiple accession, it can be multi-allelic. So now I will not explain this so detailed. But for SSR genotyping, mostly we are using this fluorescent eye-leveled genotyping tools by which you can do multiplexing of several color, dye, and you can high throughput genotyping can be done, and precise allele sizing can be done. More less than 2 base pair you can difference also. You can get that cannot be done in any other system. That's why it is the more acceptable system for allele mining in case of SSR. And SNP, as I have told, so there is several advancement has occurred from clone sequencing to this is medium throughput. Now we are going to GBS. And also next generation sequencing approach. So that is several things. Some of the example I will give quickly. One is mass array. So that is, we know Agena platform. So where you can label this SNP based on mass difference and time of flight, you can get a difference. So it has some medium throughput application, and also insertion deletion can be validated here. And another approach, Casper assay. So it is mostly utilized for foreground selection for less number of SNP. You can use in large number of lines, so that it can be, and also for DNA fingerprinting, molecular barcoding. So this kind of platform one can use. And later on, this high throughput for gauge analysis, it has come to 2 kind of platform, that Illumina Golden Gate assay and also Illumina Infinium assay. So where you can do up to 6,55,000 SNP multiplexing. And in 2 days, you can genotype up to thousands of accessions quickly, so that all genotyping data you can get quickly. So, but mostly these are mostly based on known SNP information. So you know to first SNP position and their flanking sequence, then you can design 3' and you can genotype. So therefore, only known SNP, once you mine the new allelic variant, so that can be genotyped using this technique. And also you recently, as you know that, so there is 50K array, 700K, and we also developed 90K arrays that I will explain later. So you can see here this bead can be take up to 50,000, like 700K, 700,000 SNP can be put here, and it can be genotyped in the 96 plate. So all 96 sample can be hybridized at a time. So it is in solid phase as compared to this Illumina. So it is in liquid phase. So therefore this is more highly reproducible for genotyping. That's why for known SNP genotyping, so where the SNP was known, and you can design primer. So this array-based genotyping is the most acceptable. But how you can go for new SNP, for novel SNP? So where mining and genotyping both can be combined. So that is the new technique that already we all are using. That is GBS approach. So where in Illumina HiSeq you can be done. So where 384 genomic DNA can be digested with APK1 enzyme, and it can be pulled through barcode. So once it is ligated, it can be pulled and sequenced in the one sequencer, one lane. Once it is sequenced, so then you can demultiplexing them utilizing the barcode. So based on this Illumi barcode, you can demultiplex and you get sequence data. So therefore, in low coverage, you get sequence data. But major problem in this is the non-uniformity of data, because you are getting only 0.5x, 1x sequence coverage data for one SSN. So therefore, more multiplexing, more problematic. So therefore, one has to optimize that so that you can get uniform data. Otherwise, if you compare 2 SSN, you get 2 lakh SNP. If you compare 96 SSN, you get only 200 or 200 SNP. So therefore, people have shifted to whole genome resequencing, because continuously, once the third generation sequencer has come, this Illumina short read sequencer price has reduced. So therefore, people have directly gone for sequencing instead of restriction-based RAD sequencing or GBS. So where that non-uniformity of is there, so there is already explained this 3000 rice has sequenced in the 14x coverage, so 10 to 15 GB coverage, and also get 18.9 million SNP. And also in case of chickpea, so that is already explained by Professor Bansal, so 429 previous year, now 356, and same way. So once you genotype, you get several kind of sequencing data and that allelic variation data, and you can understood their diversity pattern, domestication pattern, and also trait association also, for which trait once these SSNs are phenotyped. So coming to our research, what we are doing at NIPGR based on this concept, so that I will explain so that you can see how you have allele mining can be useful for crop improvement. So that we have already demonstrated in our research. So basically in our lab, we are mostly concentrating on yield and productivity enhancement of chickpea. We try to improve yield and productivity in chickpea in both ways. One is we try to improve high seed weight and seed number per plant. Another way, we try to increase the planting density without compromising the seed size and seed weight in chickpea. Because chickpea is mostly spreading type, so we try to make them erect or semi-erectile so that more plant can be accommodated in the less cultivable land areas. In that way also one can increase. So therefore, we are trying to understood these things. So as you can see here, so genes, we are trying to see superior allele, which is better. So I will not explain this so detail, because as you can, next slide you can understood. So likewise, what we have done, we have taken the Icrisat germplasm. So that is, and from there we constitute a association mapping panel based on trait of our interest, like seed size and seed number, with the help of the Dr. Upadhyay. So, and also we generate several kind of mapping population. So both we phenotype them, genotype them, and also we do GWAS, QTL mapping, and identify the superior allele. Likewise, I have explained, and we try to progress in the both way, marker assisted selection and genome editing. For association panel, we utilize 557 SSNs, and for different kind of mapping population for several traits we develop, and also we have mutant population. So likewise, you develop mapping population, and this is the glimpse of seed size and seed weight variations, and these lines all are phenotyped in the several geographical locations, Icrisat, IRI, IIPR, and NBPGR, and several locations, and they are phenotyped for not only for this yield trait and several other traits also, and we included wild species, Indian variety also, to check that whatever allelic variation or new allele we are mined from the land races, that is mostly in the Icrisat germplasm, so that is really present in our variety or not. If it is not present, then we try to introduce. Or in wild species, how they are domesticated. So that's why for comparison purpose, we include both all these, and we phenotype them, and you see their variation in case of seed size, seed weight, and mostly we try to dissect the seed weight. We say that it is highly heritable, so in several locations and several years. Then we try to genotype them through genome resequencing, mined 6 lakh SNP, out of which we used only 83,000, which are present in all SSNs for genome-wide association study. And this is the basic step for understanding the genome-wide association study, to understand the diversity so that we get the best loci to be associated, non-spurious, to remove the non-spurious one. So we done population structure, PCA analysis, and then we done the association study. But based on association study, we get one SNP in the ABC transporter gene, and we try to validate them, putting more SNP through gene by gene resolution association and analysis, not only GWAS, and we put that, and both we get this gene which is associated with the seed weight. So then we try to know that actually this is seed size weight related, or this is due to diverse seed size weight panel we have taken, that's why it is coming. That's why we try to validate them through QTL mapping. So therefore we taken one Desi low seed weight, high seed weight, Kabuli low seed weight, high seed weight, and we generate two mapping populations in a real mapping population. Then obviously we phenotyped them, genotyped them, and construct genetic linkage map. Once we constitute the genetic linkage map, we get the QTL, and this QTL region we try to narrow down utilizing several strategies. So then we narrow down this QTL to 0.6 centimorgan, where we get 6 centimorgan, and it is corresponding to 17.2 kb in the genomic sequence, where this CABCC that we have already got in case of GWAS, the same gene was lying, and we see that the SNP which are linked to this trait, it is giving 0 recombination. So therefore it is the similar SNPs are linked to the trait. So therefore we try to see that the same ABCC gene is maybe responsible for seed size and weight. So therefore we name them as CABCC3 based on genome-wide characterization. In chickpea there is 139 ABC transporter gene. We try to know which ABCC is this. So we name them CABCC3, and we then try to constitute the haplotype. So this gene we can sequence in several 291 accessions of chickpea and constitute 3 haplotypes that I already told. We need to identify the superior haplotype so that you go for introgression purpose. And it is mostly present in the North Indian chickpea variety. And we try to utilize this haplotype from the donor to backcross and constitute the neoisogenic line. So once this neoisogenic line for low seed weight Desi line, low high seed weight Desi line, low seed weight Kabuli, and high seed weight Kabuli. So we see them. So this line is introgressed, and it was compared with the already available variety in terms of yield and other things. It is giving pleiotropic effect or epistatics effect. So it is giving higher yield also, and it is also high seed weight also. Then we try to do molecular biology, how this ABCC transporter gene is responsible for giving high seed weight and high. So therefore we can see this gene is giving higher expression in the cotyledonous stem in the high seed weight type, and we try to do northern analysis, and I not go into detail. We do several analysis, the transporter assay to see that how it is transporting and the gene localization, all these things. But coming to the summarize of this work, you can see there we can this 291 ICRISAT germplasm, biparental mapping, GWAS, QTL mapping, fine mapping, we got the gene superior haplotype. So once we got the haplotype, we try to understand their function. So based on their function, you can see it is transporting glutathione conjugate. Due to transport of glutathione conjugate, it break down the sulfur-rich compound. Therefore, not only seed size, it improves the protein content also. Therefore, this is the line which is Ethiopian origin. It is lying in the ICRISAT gene bank. So that already emphasized by several speakers. What is the relevance of genomic and phenotypic characterization of germplasm accessions lying in the gene bank? As you can see here, so this line has the donor. So you can introgress to JG11. So this is the high yielding variety to get their yield and productivity and protein content. So it is now in the ICR varietal trial in the ABT2 to see their impact. So in also similar way in Kabuli also. So this is the Mexican origin rice. So a chickpea which introgressed into CAF2 to develop improved CAF2. So that also extra large Kabuli type trial we try to put. And this is the All India coordinated trial data. So it is giving also fusarium yield, all disease resistance also, and it is giving higher yield. It is the ABT1 data. And this line was already enlisted in the DBT biotech intervention towards self-reliant India. It's released by Honorable Prime Minister. And likewise, we try to develop several kind of strategies, so that I not go into detail due to lack of time. So that this QTL region, how you can put several kind of NGA strategy that we called as a QTL region specific association analysis and get to the gene instead of hide mapping or generating more mapping population, how quickly this QTL can be narrowed down to the particular gene. So that kind of strategy we have developed through genotyping tool. and it was published in the DNA research. And also, as we now already people are using that, we know QTL-seq. So where we develop another approach that multiple QTL-seq. So where one of the parent is common in the two mapping populations, so that both extreme type you can isolate DNA, pull them and resequence them and get the QTL. So once you get the QTL, the both mapping population QTL, you can validate each other, so that multiple genetic background validation at a time you can do, and in the process you maybe try to narrow down the gene QTL region to the particular gene. And likewise we have tried for podding, flowering time also, and also like GWAS QTL mapping. I have already told, putting functional genomics, we try to delineate the genes for flowering time and several new allelic variant for early flowering, and this is the known gene and new gene also, new allelic variant also. So similar approach we do. And another I have recently we have developed targeting the shoot apical meristem of the chickpea. So where you can see the clavata gene, which is the master regulator of shoot apical meristem regulator. So this CACLV3 can be regulate flower number also and early flowering also. So that we try to dissect through this. I have already told the similar approach. We get the haplotype, understood their function and introgress and develop this line. And also recently, this was the erect and semi-erect type also. This is the also through shoot apical meristem dissection, we can identify the USEL gene. So how it is regulating, so that I will not go into detail, but this it is giving, increasing the planting density and productivity. So basically our plan is to implement the genomic assisted breeding strategy, association, QTL mapping, map-based cloning, or all functional genomics approach, understood the gene function, identify the new allele, then in progress, and go for their validation either through genome editing or through marker assisted selection. So likewise for drought also we have recently identified. So because of lack of time, I'm not going into detail, and this is the seed color also. The similar way we identify new haplotype. For several color, I can tell which haplotype is responsible for which color. So all this has been recently released by DBT1 book, the cutting edge technology in impacting agriculture. There it is enlisted. So likewise, we have dissected several QTLs, genes, a new allelic variant for not only for agromorphological trait, seed quality trait, and also for photosynthetic efficiency, drought, vernalization also recently we have done. And we published also, but recently we try to put all these traits together to put in the JG11 and this PUSA 362 to make is a kind of gene pyramiding work. So we try to initiate that, and recently we are working on this chickpea mission project, and Dr. Chaturvedi and Dr. Bhetri is already here, so they are the part of this project. So, and also RLBCAU is the part of this project. So I need not to explain this, because here one can see what is the importance of germplasm accession. That only we try to do here. Germplasm accession, we try to do genomic characterization and phenotypic characterization, and identify the QTLs genes for several biotic, abiotic, mechanical harvesting, and yield traits, so that we get best loci to be introgressed by best allelic variant or best haplotype and best donor for introgression purpose, for future breeding purpose. So to expedite our breeding approach, recently we developed in the rice this 90K, 80K SNP array data. So where this pan genome SNP is put, not only that chickpea, rice, Nippon bear genome that I have already told, OS, Basmati, all SNPs are there, and it is widely utilized by several rice breeder recently, and it is also now utilized in mission mode project of germplasm characterization in 15,000 rice system that already has explained by Professor Bansal, that 15,000 already has been done, and that also we are utilizing here for genotyping, genotypic characterization. This array will utilize, and this was already patented, and recently it was released by Honorable Science Minister in and previous secretary. So in October 8 for national use, and likewise we have developed several databases so that quickly breeder can get this SNP for several traits. So like rice. So here one can put their trait, you can got which loci to be associated quickly, so that So we can go for trade interrogation. So as I have already explained, so through Allele mining, what we can do, so we can improve the trade. And this is the roadmaps, as we already know, from traditional breeding to now we are coming to sequencing them all. So I need not to explain this. And there is several problems in this, I know that. So we all know that the computational tool, the sequencing, as I told, it is not easy, because once you have sequenced that of 1,000 germplasm, how to tackle that, how to do downstream analysis. So therefore, DBT has put this facility, this NGGA facility anchored at NIPGR, so where this kind of work can be done, and whatever platform, genotyping platform I have already explained, Gene Titan, that Illumina, all these are there. So one can come here and do work, and also downstream analysis that scientists can help them. So that, and also the consultancy also, which platform is important for which trade dissection, because for one SNP, maybe 1,000 germplasm, maybe sequencing will be not the right way because it will be more cost intensive. So maybe Cas-Space will be, multi-top will be good. So that kind of consultancy can be done from this. And at last, I will acknowledge my all my lab members who are working, and also the group member of the Chickpea Genome program, and also all my collaborators from ICRISAT, IRI, LBPGR, and also Professor Bansal, who is here. So when he was director, he has helped me at all the germplasm from NBPGR, and also Dr. Chaturvedi, who is helping us now for all this, and also all this chickpea work. I used to get his suggestion from Shailesh Tripathi sir, from him, because they are also guiding me for doing this work, and also in chickpea mission program also the similar work we are doing. The what is the webinar title today's thing only. So one, because basically our approach is to do allele mining. So that is the main approach, utilizing the germplasm accessions, genotyping, phenotyping, identify the new allele or superior allele, which is associated with the best trait, and we try to identify the donor and loci to be introduced so that anybody can utilize in the future breeding program. So thank you all for giving me this opportunity, and if you have any doubt and anything, so you can ask me. So I will be happy to answer that. And thank you very much.
Dr. Vanu, please unmute yourself. Dr. Vanu, you are not audible.
Dr. Nisant, we can't hear you. First unmute. Yeah.
Yeah, unmuted, but some problem is there, I think.
Dr. Nisant, is there any problem? Dr. Manit, you can Dr. Manit, you can proceed further.
I think Dr. Manit is also not here. So yeah. Now
I think let me. I think it was an excellent talk. Congratulations to Dr. Prida, and again thanks to you for acceding to our request to deliver lecture. And because I was expecting that a lot of information you shall be sharing during your presentation, and same thing you did also. I think many students are also there, and they will certainly have a closer interaction with you later, because as discussed earlier, we shall be deputing maybe 3 students for 1 week training with you. Maybe this is sometimes in July, or maybe in June, July. So That's right. Otherwise, it was excellent talk. Thanks to you. Thanks to Professor Bansal sir. I can see him. Good. So if there is any clarification, any question, any query from anyone, please, Dr. Parida is here. Please.
I think there are not many questions today. So
am I audible now, sir? Okay. Am I audible, sir?
Yes, Dr. Nissan. Yes, audible.
Okay. Okay, so thank you, sir, for highlighting the importance and adaptation of this new technologies, this span genomes, next generation molecular breeding, and its role in advancement in crop improvement. We're highly obliged to you. And I am also very much happy to inform you all that altogether more than 300 participants have registered and witnessed this webcast through Zoom, YouTube, and other social platforms. From this webinar, we have come to know many scientific developments in the field of crop improvement, germplasm characterization, genomics, allele mining, and the role of modern tools. I trust that the knowledge shared by eminent speakers will help us to design our research programs and to achieve our scientific goals. I once again thank all the guests and speakers. In this continuation, today, we have with us our Honorable Vice Chancellor and the Chief Patron of the event, Dr. Arvind Kumar. He hardly needs any introduction. He's a man of distinct vision and the fountainhead of illuminating ideas, an idol of knowledge, experience, and inspiration to all of us. Sir, we admire your spirit of integrity with 100% dedication to the world of research and education. You are an inspiration to many of us and will continue to remain so. On this occasion, I request you, sir, to grace this occasion with your presidential remarks. Sir, if you are here, sir, Vice Chancellor, sir.
Hello. Yeah, Dr. Nissan.
Yes, sir. Please.
Dr. Kalyan is not here?
No, sir. Sir, he is not here. Sir, he has some emergency, so he was not able to attend this program.
That's what I was waiting, that he must be.
He's having some medical emergency, sir, actually.
Hello. So I think this is, we have come to the close of this national webinar on new dimensions in crop improvement from allele mining to genome editing. I was listening to various speakers and I was really very happy to listen to all the learned speakers who have spoken on a wide range of subjects, how we can make improvement, particularly the crop improvement, what the role of genetic diversity, genomics, how we can have genome engineering, and also about the plant genetic resources, next generation molecular breeding, and how we can make crops nutritionally rich, healthy, and strong. So various topics were quite relevant, and I'm sure that the participants must be certainly benefited with the just a minute. नमस्कार जी, मैं 2 मिनट में भी बात करता हूँ, just so I really thank all the speakers who have devoted a lot of time and giving their valuable thoughts on this very key aspect. And I'm sure that particularly when Dr. Bansal was speaking, he spoke about how the germ plant played a very vital role in the crop improvement. And particularly, it was also interesting to note that in India, the utilization of the germ plant as far as the CGIAR from the CGIAR system and vis-a-vis the China, who have their own resources they have exploited. So this kind of, indications gives us enough thought that how we can improve our genetic resources, how we can utilize our local cultivars, how we can make, the germ plant available in different agro-climatic zones for the benefit of crop improvement. So this kind of, and I have seen that how the wheat germ plant, particularly in Haryana, the pulse germ plant, and this is really a daring step. It's not, possible to expose such a large germ plant to public, but this was a very significant achievement. And through this, lot of selections, a lot of people could see the germ plant diversity, utilize it. So ultimately, it rests how best you can collect, how we can characterize, how we can utilize all the germ plant resources. And our country is fortunate to have a very strong gene bank. And NBPGR has always played a very significant role, very key role as far as the repository of various bioresources in India is concerned. I could also see that in India, this is the, ultimate breeding approaches and the molecular approaches. In recent years, the biotechnological tools have given a new shape to the next generation breeding, I call. And ultimately, we are much wiser to understand the plant systems, as it is very clear that the sequencing of genomes in different crops, it is available by the characterization. We understand what kinds of genes are involved in a different kinds of stress and so on. So all this information was not existing earlier, and it was only through the traditional approaches of breeding. Now, with the advancement of science, with the advancement in biotechnological approaches, there is a lot of, growth as far as the varietal development is concerned, as far as the nutritional quality is concerned. And you know very well that about, 3 dozen varieties have been released through biofortifications, and these are nutritionally superior. So this has become possible only by understanding our, plant system, what is happening, what are the genes involved, what is their characters, how best we can utilize in our breeding system in different agroecological regions, like the salinity was being talked about, and I am very happy to see that the President Award has been given to Dr. Parikh. And not only Dr. Parikh, all the scientists who spoke today, they are all very learned and are very well, recognized in the scientific arena, and all are, have played a very key role in the overall improvement in the, crop improvement, and particularly the understanding of the system. Now, with this, it was really pointed out that our land is shrinking, and per capita land is declining, the population growth is there. So really, these until unless we make the new advancement in science, we understand, we utilize it under different biotic and abiotic steps, it is not possible to make any further improvement. And the yield decline, there is one potential, productivity. Another is actual productivity. There is a war gap between the potential productivity, actual productivity, and the farmers level productivity. So if we are able to see what are the reasons why this potential productivity is less, and how we can narrow down the gap between potential productivity and actual productivity, that is the future approach through which we can further bring additional production in our kitty. Now it is again very heartening to note that this year, because of the advancement of the science, because of the new varieties, new technology, the country could produce 316 million tons of the food grains. So this is a very remarkable achievement. And in fact, while I was looking the second estimate, it is very apparent not only in food grains the country could have a record production, but in rice 127.9 million tons, wheat 111.32 million tons, maize 32.42 million tons, pulses all-time a record production of 26.96 million tons, particularly in chickpea gram, which is contributing almost half of the pulse production, 13.12 million tons. In oil seeds, rapeseed and mustard, 11.46 million tons. Sugarcane, 414.04 million tons. And jute, again, a very high production. So, particularly in rice, wheat, maize, pulses, chickpea, rapeseed and mustard, sugarcane, we could have a all-time record high production. So this has become possible only through the leading varieties suited for different biotic and abiotic stresses, through better understanding of the plant system, through the better biotechnological approaches, these molecular tools, which has ultimately made possible to breed such varieties which are having more productivity and having better tolerance to various abiotic and biotic stresses. The country needs, to feed ever-growing population, and soon across the world we would have 9 billion population. And you know very well that India is a country which supports, about 1/6 of the world food production. So the country, India, has to take lead, and in that direction, the biodiversity in India at different places need to be further conserved, which need to be, further utilized and value added, because the local types, local material, we can take lead from those local material which are growing since ages under those agroclimatic situations, and ultimately upscale their values, their traits, so that we can boost up the productivity at the national level. So I am very happy that this particular seminar has been organized today on the occasion of the Science Day, and we are celebrating Science Week. We have our Foundation Day on 5th of March, and I am happy that this time we are celebrating physically, and because for the last 2 years we do have the corona pandemic, and therefore we couldn't meet physically. So this time we are trying to meet physically, and I'm sure that the inputs which have been given by various speakers, including Dr. Chaturvedi, Dr. Bansal, Dr. Parikh, Dr. Mani, Dr. Parida, these are very, very significant and pertinent in the present day context. And I am sure that our PG students must have really been benefited, and they must have noted the various points. And as indicated by Dr. Bansal, he had correctly pointed out that the students must give a feedback, what they have learned out of. So if they give the feedback, their suggestions, that will be very valuable, so that we are able to understand how much the students could gain from these very important topics of learned speakers from various parts of the country. So with these words, I again thank the organizing team for organizing this national webinar on this Science Week. And I am sure that such types of the interaction would further continue, which shall further pave way for encouraging our students and faculty to take the new ventures, new areas, new innovations in science and technology. Thank you very much.
Thank you very much, sir, for your speech. We are grateful for the valuable words and efforts you took to share your thoughts and experiences. So now, for the formal vote of thanks, I would like to invite Dr. Piyush, the co-convener of this national webinar. Dr. Piyush, over to you.
Okay. Thank you, Dr. Nishant. So at the end, I'm happy to say a vote of thanks. So start with Honorable Vice Chancellor, most valued invited guests and respected deans and directors. A very good evening to every one of you. It gives me an immense pleasure to deliver the vote of thanks to all the dignitaries present here for this webinar. On behalf of Rani Lakshmibai Central Agricultural University and the organizing team of this webinar, we would like to thank our distinguished guests, Professor K.C. Bansal and Professor Ashwini Parikh, who honored this program by delivering valuable lectures and thoughts. We would like to thank our Honorable Vice Chancellor, Professor Arvind Kumar, for gracing this webinar. Sir, you are always a source of inspiration for all of us. Thank you very much, sir. We are grateful to other speakers, Dr. Vetri Venthan and Dr. Parida, for demonstrating their research on this important topic of crop improvement. Thank you very much, sir, for your lectures. We extend our thanks to Professor Anil Kumar, Director Education, and Professor S.K. Chaturvedi, Dean Agriculture, for their valuable thoughts and support for making this event successful. We would like to thank all the Dean and Directors of our university and the Directors of IGFRI and CAFRI, two of our neighboring institutes. We also express our sincere thanks to all the faculty and staff members, students, participants, and audience for making this event successful. We also thank our sponsor, NAHEP and Agrivision for you. Last but not least, I extend my sincere thanks to organizing team, Dr. Ansuman Singh, Dr. Manit Rana, Dr. Nishan Bhanu, Dr. Tanuj, and Dr. Selen for their cooperation in the organization of this webinar. I acknowledge their contribution to make this event successful. Once again, I thank you one and all present here. Thank you very much. So before we wrap up, I request all of you to stand up for national anthem. Dr. Tanuj, please.
Yes, sir.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जन गण मन अधि
Thank you very much once again to every one of you.
Thank you. Thank you, Manzal Saab.
Thank you, Dr. Piyush Allele.
Thank you.
Thank you all
sir.
the speakers today. Thank you.
Thank you, everyone. Thank you. Bye-bye.
Dr. Mani, bye-bye.
Thank you, sir. Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Excuse me, sir.